confirming that it's live, and I don't have a URL. You're looking at our YouTube page then?
I'm just checking.
Here it goes. Preparing to live stream the meeting.
Ah.
Hi, Dr. Massa.
Hello, hello.
I think she's still working on setup, which is great.
Yeah.
Hey, Mike.
Good morning.
Hello, hello.
We're gonna do,
Hey, Mike.
Hi, Dr. Mousa. Can you hear us at all?
I think she dropped out. Oh, she's back.
It's your video. She's not muted. Mic is muted.
She's
Maybe
working on it. It's
you-
cool.
Hi,
So
Dr.
it's
Mousa. Can you hear us at all?
she dropped out. Oh, she's back.
No, she's here.
It's your video. She's not muted. Mic is muted.
Yeah, if you can do me a favor right now,
Hi,
guys,
Dr. Mousa. Can you hear us at all?
She dropped out.
can you put your YouTube on mute?
Your video. She's not muted.
Oh, it's me.
If
Hold
you
on.
can do me a favor right now. We got the loop going there. Thank you, Tony. So we do have a YouTube channel now that's live. Do you see that? Or do I need to publish that to you, Tony? I'll put this in chat. So Dr. Massa, we're connected to YouTube, but it's not a published channel yet. And we're going to be pushing to Facebook. So we might start a minute or two later, if that's okay.
Of course.
I'm so glad you could join us.
Oh, thank you for inviting me. I wasn't sure if you wanted me to present anything or if we're just going to have chats and discussions.
Well, you have so much knowledge and information. I think if you provide an overview, and our close team here might have some questions, and then we're going to be monitoring questions on Facebook and YouTube as well. You might see some other teachers
Wonderful.
popping on as well here, but most of the
Okay.
conversation is going to be outside this channel.
Great.
I was going to send this to Tony. So we're working through the logistics here. Here is, and feel free everyone to share this with your networks. This is going to be pushed into our Facebook page. Tony, if you can do that. We're right kind of on the top of the hour. So I think we're good for that. We are on YouTube live, and Tony's setting us up on, Tony, if you can just give me a thumbs up that you're going to set this up on Facebook. I'm going to get started. Oh, you're on mute, so just yep. Awesome. Well, I'm going to get started. Well, good morning, everyone, or good afternoon if you're on the East Coast, or good evening, or good morning if you're in different parts of the world. I'm Ted with Magnitude, and this is session number 4, Magnitude Live. Our world is a little uncertain right now, and we're trying to create a little more continuity and maybe some fun and enjoyment as well, and take you a little bit beyond Earth and look back at our planet. And we're doing some amazing things in orbit. We've had something operational above our planet for 2 decades, and they're doing absolutely amazing stuff. We have projects we send up there. It's called ExoLab, in which we look at seed germination and plant studies in this environment. But there are scientists, engineers that are doing far more advanced work, and we're encouraged by what they're doing. And we're really fortunate to have one of the lead scientists in the world really studying, I guess, astrobiology is probably a good way to think about it. What will our astronauts eat when they're on the space station? What will they eat if we get them on the surface of the moon or on Mars? And so Mary, thanks to Mary Vaughn, one of our teachers in Florida, she has a great connection with Dr. Massa. So Mary, maybe provide an introduction, introduce yourself, and share our guest with us.
Hello, Dr. Mousa, and welcome. We are so glad you're with us today to help us inspire our students. So Dr. Massa is one of the chief scientists with Veggie. She works out of Kennedy Space Center, and her team works, and has been working the last several years with growing items on the space station. So without further ado, I am going to turn it over to Dr. Massa. And if you want to tell us about, what you have done and what the future missions may incorporate with all of the growth items that you have done. So thank you for being here.
Great, Matthew. It was a pleasure to be here, and thank you, Mary, for inviting me. I apologize. My husband's also on a conversation, so you may periodically hear Italian coming in through the microphone. But we're all experiencing interesting times working from home. So thank you for the great introduction, Mary. As Mary said, I work with a project on the International Space Station called the Veggie Project. And so Veggie is our small plant growth chamber that we have on the International Space Station. We actually have 2 Veggie units on the space station right now. But, first I want to talk a little bit about why we're interested in growing plants in space at all. So plants are fascinating. I love plants. I'm a plant scientist and I've studied plants for a long time. And, a lot of people don't really think about plants very much. So why do we grow plants in space? Well, we grow them, first of all, for the food. Plants are going to be a source of fresh produce for the astronauts. But there are some other potential benefits of growing plants. Plants could be a very good source of a psychological connection with Earth. Okay, so if you're living and working in an extreme and stressful environment far away from home, having something that gives you that connection with what you've always known is going to be very important. And I think that, watching something grow and develop over time, like anticipating when you're going to be able to harvest and eat that, I think is going to be really key for the astronauts. And we're actually doing surveys with the astronauts on space station right now to try to get to how important or what part of their lives plants will play a role in. Plants do other things too. So at the same time that your plants are growing and producing food, they're also generating oxygen. They take up carbon dioxide from the atmosphere, and through the process of photosynthesis, plants split water and generate oxygen that the astronauts can breathe. So they're helping to recycle the atmosphere and purify the air. So this is another really important role of plants. And a fourth role that I can see plants taking in future life support systems is water processing. So plants take up water through their roots and they give off water vapor through their shoots. And this water vapor is actually totally pure water. So it can be collected and condensed and reused. So plants could have a whole lot of roles. And I think what we're doing on the space station right now is really starting to investigate those roles, starting specifically with the food production and the fresh produce to supplement the packaged diet. So the packaged diet that astronauts have in space is really quite good. And when we look at the food that the astronauts eat, it's pretty diverse. There's a couple of hundred of food sources available. Things like shrimp cocktail, vegetables and fruits that are freeze dried or thermostabilized. Things like tortillas that they can use in place of bread. So there's quite a lot of food, and it all has to be very highly rated. But over time, if you store that food, the quality and the nutritional content of that food will actually degrade. And so this isn't an issue on the space station because we're sending them fresh, new supplies of food all the time. But when we are on our way to Mars or to the moon, that could be much more of a problem. If we have to send the food ahead of the astronauts to the surface of Mars so that the food that they need when they get there and when they return to Earth is waiting for them, it may have to be there for quite a long time. And for this reason, we aren't sure we can guarantee a complete diet for the astronauts. So we're really looking at being able to provide fresh produce to make sure that they can get all of the vitamins and nutrients that they will need for these longer duration missions. And it also will add some variety to the diet. It will give them, new types of foods that they can create. So the veggie chamber, which you're seeing photos on the screen here of, is a small astronaut garden that has been built and sent to the space station. And it's got these plastic accordion style bellows where you can kind of collapse the whole thing down when it's not in use. And then you can expand it to be able to grow the crops. And you can draw the bellows base plate away from the light to keep pace with the plants as they grow. So as the plants get bigger, you can lower them farther and farther down. We have LED lights in this chamber, red, blue, and green LEDs. And, you know, part of the reason that we have those colors is because the red and the blue are really important for photosynthesis for the crops. The green are really important for the crew to make the plants look like plants, to make them, you know, give them that psychological appeal, but also to help with diagnosis of, you know, is this plant healthy? Is this fruit ready to be harvested? You know, having that green component is really important for human eyes to sense what's going on with the plant. And the plants right now in veggie are grown in a solid substrate in these packets called plant pillows, which are essentially space grow bags. And so veggie was designed at a time when I was talking about the LED lights. Now we have a lot of white LEDs. They may be very good for growing crops, but we haven't actually done that much testing with them yet. But since veggie was designed several years ago, those weren't available at the time. And that's why we've really got the red, blue, and green LEDs in veggie. So yeah, here's a wonderful shot of Scott Tingle showing the variety of produce that could be grown in the 2 veggie units. So here we were growing leafy green crops, and that's mostly what we've grown in veggie so far. We've done a number of tests with different types of leafy green crops, red lettuce, green lettuce, Chinese cabbage, Mizuna mustard, which is a nice spicy mustard variety we've grown quite a lot of. We've also tested some new crops, including the red Russian kale, dragoon lettuce, a small butterhead lettuce variety, extra dwarf pak choy, and one that I think was kind of a crew favorite was the wasabi mustard, which has a spicy wasabi flavor. They said that it was really delicious with their shrimp cocktail. So the strong flavors could actually be very beneficial in space. The astronauts experience what's called fluid shift, where when you're in microgravity, your heart is still trying to pump up the blood from your feet, but gravity's not pulling it back down. And so in effect, you see astronauts, they have kind of a puffy face when they're in microgravity. And this is due to that fluid shift. And several astronauts have said that it's kind of like having a head cold, where you've got sort of this sort of pressure on your sinuses. And so your sense of smell and taste can be kind of muted or reduced. So they really like to have strong flavors. They eat a lot of hot sauce. That I think is why the wasabi mustard was pretty popular. They also love to smell the plants. So we've heard time and time again from the astronauts how much they love to kind of stick their head in, you know, near the veggie or just smell the green, fresh plants when they're up there, because it's so different. You can see the rest of the environment on the space station is very much a combination of cables and plastic and metal, and it's all synthetic. So really having plants there is one of the only natural things in that environment. And it's very comforting for a lot of people to have the sights and smells, you know, associated with Earth and with being outside or in the garden. So we're still continuing to do a lot of testing in veggie. In this photo, you can see a new watering system that's actually being tested on veggie right now as well. This was the Ponzo 2 experiment. Yesterday, they started the Ponzo 3 experiment on Space Station, where we have these more rigid growing structures. And so we're looking at trying to reduce the amount of time it takes for the astronauts to water the plants, because watering is a big challenge. Watering is actually the biggest challenge we have with growing plants in space so far, because water and air don't mix well in microgravity. And so you'll see water forming big bubbles. And you'll see if you put air bubbles inside those bubbles of water, the air just kind of clumps in the center, and water and air don't mix. But plant roots need both water and oxygen. So getting that combination of water and air in the root zone is something that we're working really hard to try to do. We're also looking at things like developing future water delivery systems that will not have to have the astronauts hand water them, because as we want to grow more plants for longer duration missions, we have to have more automation and more maybe robotics helping us out. We need to be able to do things like detecting plant stress before it becomes a problem, so that we can intervene and have healthy plants. You know, when plants become a critical part of the astronaut food, it'll be really important to make sure that they're healthy and productive. In addition to leafy greens, you just saw a photo, I think, of some dwarf tomato plants. We're hoping to grow dwarf tomatoes on the space station next year. We're also looking at chili peppers, and we're hoping to grow those this summer in another piece of hardware that we have on Space Station, which is called the Advanced Plant Habitat. We have grown the zinnia flowers on Space Station, so they were great. We had a lot of problems with the zinnias at first. We had some water problems due to the fans being shut off, and we didn't realize that. But in space, you have no convection. And so heat doesn't rise. Air will not move unless you have fans moving that around. You have no natural convection, I should say. So we have to have forced convection. And what happened with the zinnias at first was the fans were shut off, but the sensor indicated that they were on. So we didn't realize. And more water was building up, and it ended up causing the death of a lot of the plants. We had some fungus that were growing on some of the plants. That was a big problem. But Scott Kelly, the astronaut who was running that experiment, did an amazing job, and he intervened. He removed the sick plants, and he took over, turned up the fans, and was able to nurse two plants through to flowering. And when they started flowering, they just produced a profusion of flowers, which, you know, the variety of zinnia we used were profusion zinnias. So here were the first flowers, but they actually continued to flower and flower and flower. And we ended up getting about 28 flowers from those two plants. So yeah, Scott took beautiful photos of the plants. He took the plant pillow out of its it was bungee corded into the veggie, and he took it all over the space station and took some gorgeous photos of the zinnia when it started flowering. And one of the coolest things he did when he harvested was he asked if he could make a bouquet. And we said, yeah, of course, you know. And so he made a bouquet of the zinnia flowers at harvest and took photos of that as well. And that was just really, really powerful for our team. It was also really wonderful to see art created in space, you know, by something that we did. And that's really another area that plants could potentially be a benefit to the astronauts. You know, it gave them this opportunity to create art and to have this beauty in their environment that, you know, that they might not have otherwise had. So that's kind of a high-level overview of what we've done so far. What we're looking at in the future are the development of space crop production systems that will help to really enable humans for their different exploration missions. So we'll probably have different missions when we go to, you know, the moon, to go to Mars. We'll have to have different crop production systems for those different environments. some of the situations might require really short growth periods. So we're doing a lot of work in an area with microgreens. So microgreens are 7 to 14-day-old young plants that you can grow in a very dense plant canopy, almost like a lawn. And then, you know, I make the analogy, you grow your lawn, you mow the lawn, and you eat the grass clippings. It's the same idea with microgreens. You have a lot of seeds planted. They grow up into this very dense canopy, and then you can just mow them off and eat those young plants, and they're very nutritious and they're very delicious. So this is one way that we might be able to supplement the crude diet when we only have short durations. And then for longer durations, we'll be growing more and more salad crops, so leafy greens, tomatoes, peppers, and we're looking all the time at new crop candidates. We have a lot of help with that. We have middle schools and high schools around the United States helping us to do crop selection through a program called Growing Beyond Earth, which is run out of the Fairchild Tropical Botanic Garden in Miami. They're generating data, and the data come into NASA, and then we find candidates that grow very well in a lot of different places. If you can grow a plant in Puerto Rico and a plant in Colorado and have different students taking care of them in the different locations, and they grow well in all of those different locations, then that plant's a pretty good candidate for space. And so we're using that to help feed into our crop selection process. And then we are identifying which crops we want to grow next by growing them under high fidelity conditions at Kennedy Space Center. So we have a lot of help from schools out there as well, doing a lot of fundamental experiments on plants in space to give data for what might germinate, what might grow well. And so we really appreciate you all and your group doing a lot of this work and helping to generate those data, because there are a lot of questions that remain, and we have to figure out a lot of solutions to be able to enable astronauts to live and work all over the place. So thank you all, and yeah, I'm glad to answer questions.
Oh, Dr. Massa, thank you so much. Anywhere from the psychology of having the plants on the space station to flavor and how important that can be, to thinking about water delivery systems and how really the physics on Earth are completely different when you're in this microgravity environment with water and with air, and how carbon dioxide might work, to the plants themselves and how these different stressors, whether it's gravity or the high CO2 or temperatures or whatever it might be, these are absolutely fascinating. The teachers that are joining us here directly inside the Zoom and the teachers that are joining us via YouTube or Facebook have maybe different connections to what we're doing with ExoLab. If I can, I'll provide a brief introduction to ExoLab, and you can see we have some limitations based on what we're doing and how we're doing it. And if you have any guidance or insight, we'd love that. We were really privileged last week to have Dr. Richard Barker from Gilroy join us. And he took us on a fascinating journey. It was really, it was like improvisational jazz, honestly. About 3 hours, he really went into data science and what have you. It was quite an extraordinary insight to how you might take this growing, living thing and then quantifying it, right? Getting some information about it. With the ExoLab and with the teachers we have here, we have teachers in Africa and in Europe. And I think pretty much it, well, I've got Gil's from Japan. So he's joined us today from Osaka. Hey, Gil. But most of us are here in North America and Canada and here in the United States. We're growing our plants inside this ExoLab chamber. Mary's got one behind her here. Mine's right here. I'm gonna disconnect it. And we're growing these in an agar medium. And we're growing those on the space station as well. And of course, that's great for germination, but it's really not great. Oh, there, look at Michael's Wilkinson. He's showing us the plant up close. That's a little better view. Let me just switch to you, Michael, if I can. Oh, there you go, you can see it. And so it's great for germination, but it's not really great for total view. But one interesting benefit that we're having is the ExoLabs have a camera, and it captures a time series in addition to data like carbon dioxide, temperature or relative humidity. Oh, I love when our children join us in these calls. You know, it's quite an incredible time. I'm just getting distracted by Lauren's kid that's joining us, which is fun. And so we're getting this time series macro view data of the plants, but not the micro. All right, not getting into the genetics and the epigenetics. And so I've got just so many questions, but I want to open up to the teachers here if they might have a question for you. And if C has joined us here, if you can check on YouTube and Facebook as well, we might have some other questions coming in. So guys, feel free to unmic your mic and ask Dr. Massa a question directly.
Dr. Massa, hi, my name is Mike. My question for you is, you mentioned that you're growing it in a more solid substrate. What happened to hydroponics? I thought that was going to be a big thing, looking up in the space station and things like that.
Yeah, that's a great question, Mike. So hydroponics actually is kind of a very general term that encompasses anything where you're growing, without soil, and you're growing using nutrient solution. So this could actually have a substrate. A lot of hydroponics has been done in sand and gravel. We're actually doing kind of a modification of hydroponics in veggie. We use a soilless substrate that's a baked ceramic. So you can think of it like a terracotta pot that's just been crunched up into little tiny pieces, or even like a chia pet that's broken up. And we put controlled release fertilizer in there, and so the astronauts just have to add water. It's not ideal. It's not sustainable at this point. This was really a proof of concept test for veggie. We're able to plant the seeds dry in these plant pillows, package them and send them up, and then the astronauts just really have to add water. But that's not how we're going to do things, for future systems. So we actually have a lot of work on water and nutrient delivery system research right now. And NASA just gave out grants to 5 different institutions, companies that will be also helping both with water and nutrient delivery and volume optimization. So we're going to get a lot of help from a lot of other experts. But definitely hydroponics is the way that we're going to be going in the future. And hopefully a hydroponics approach that doesn't have a substrate, or that has a substrate that can be recycled or reused. You want to take as little stuff with you as possible. And so one of the most promising hydroponics approaches that we're looking at on the ground right now is called porous tube nutrient delivery. So this is where we have a porous ceramic tube. So again, think of a chia pet, and we can grow the roots, put the seeds on the tube, and the roots will grow around the outside. And then when you harvest your plant, you're just left with this massive roots on the tube. The tube has your nutrient solution running through it and pushed out through the pores. So the roots can't go into the tube, but the solution can get a thin film on the surface. And so that also allows oxygen to get to the roots. If you just have a pretty thin film of this nutrient solution, it's thin enough that oxygen can diffuse in and be there for the roots. And that's really that happy medium that we're trying to find, where you have enough oxygen, but also your water and your nutrients. Not too much oxygen, because then you're in a drought situation, and not too little, because then you're in kind of a flood situation. And we're often falling off one or the other side of that tightrope in space. So the great idea about the porous tubes is when you harvest your plant, you're left with these mat of roots. You can just kind of peel off the roots, and then you can, wipe down the tube and restore it. Oh yeah, this is a wonderful video of Commander Chris Hadfield getting a very wet washcloth and then wringing it out. And I think if you skip ahead a little bit in the video, you'll see. I'm not sure if we can skip up. Yeah, there he's got it. And he adds water to it and gets it super saturated. And this is really pretty great, because when you see him wring out this washcloth, it takes a little bit. This is a really great video to show your students. The washcloth gets saturated, as he wrings it out, you get kind of a film of water that turns into like a tube of water, excuse me, a tube of water, and it starts to coat his hands and it starts to crawl up his hands, okay? And so if you think of that washcloth as a plant root, you can see how easy it is for that plant root to just drown in water, to not have any air coming in. So getting that film of water thin enough so that oxygen can diffuse in is going to be really important. And so, you know, a lot of traditional earth-based hydroponics just pumps oxygen into the water. You might use like a fish tank aerator or something and put little bubbles of oxygen in there. And that oxygen stays dissolved in the water as long as it's aerated relatively frequently, and it provides enough air for the roots. But in space, the air and the water don't mix really well. There's a wonderful series of videos by astronaut Don Pettit, and he shows one where he's got this blob of water and he's putting drops of air in there. And you'll see that they all form, I think I can share that, they all form like a giant shell of air. Here, let me see if I can get this up. Okay.
That the problem we're having with our agar, in that we're kind of drowning our roots, perhaps.
Let's see. So can you guys see this video that I'm sharing?
We can, yes.
Okay. So here's Don putting like little drops of water in, little drops of air, sorry, in a bubble of water. And you see what he ends up with is, it looks like an M&M, right? It's just a big clump of air with a shell of water around it. So the air just doesn't stay dissolved. It all sort of coagulates, and the water also kind of, you know, will coagulate. And so that's really the challenge that we're dealing with. We could do a lot of fun things. I encourage you to look up some of Don Pettit's Saturday morning science videos because they're just outstanding, you know. And he talks a lot about the behavior of fluids, and so it really helps to shed some light on what we're talking about here. So, whoops, where did I go?
You're with us still.
Okay, still there. Sorry, lost the window. Okay, there we go. Yeah. So, but hydroponics is definitely what we're interested in, Mike, and you know that's really what we want to do. We want something that will be reusable. You know, your agar, our substrate, none of that's really reusable, and so that's not. You know, we can't be sending all this stuff up from Earth. That's just not sustainable. So we have to become more self-sustainable. So definitely encourage your students to think about different ways to sustainably grow plants over long durations, because that's really of interest.
Thank you. That's fascinating.
All right. I know you guys have some more questions, and your students probably have questions as well. You know, one thing we've done, Dr. Massa, with Exolab is we fly a single experiment to the space station, and then it's an Internet of Things device. We network hundreds of schools together while they're growing their ground trials. So in a way, it's very similar to the Mark and Scott Kelly experiments, in which we have a Scott Kelly plant that goes up on the station, but we have hundreds of Mark Kelly plants, if you will, here on Earth. And the students look at their plants in a gravity vector, and then those that we grow on the space station. And we're looking for changes in comparison. But again, this is kind of at the macro level, not really at thinking about genetics and what have you. Can you speak a little bit to that? I think Dr. Barker last week was really diving deep in that, maybe not so deep of a dive, but help us understand the context of how you might be looking at space plants. I know that you do research not just in making sure we have some good food, but can we modify these perhaps for different habitats and different environments, or perhaps different production qualities?
Yeah, no, it's a really good point. You know, as Dr. Barker probably talked with you about, you know, there are a lot of changes that go on when organisms are in space. I mean, everything, you know, on this planet has evolved with 1G. And so when we are experiencing microgravity, when plants are experiencing microgravity, this is an environment that they've never seen before and they didn't evolve to adapt to. So plants will have a variety of different responses to this environment. Sometimes it's kind of a stress response. This is like, wow, it's new. I don't know what I'm dealing with, and it's stressing me out, right? So it's the plant version of that. Other times it might be similar to like a defense response, like I'm fighting off a disease or an insect. And so we have a lot of really amazing investigators working on trying to understand what's going on at a genetic level. I'm more interested in how we can apply that. So we are looking at, being able to produce food for the astronauts, and how can we do this best? Well, some of the plant varieties that we have grown on Earth might be suited to grow in that environment. So a lot of our testing is trying to mimic the environment of the space station in our ground chambers here, where it's temperature, the temperature on the space station is about room temperature. It's around 22 degrees Celsius. The humidity is pretty low. I mean, I'm sitting outside in Florida right now, so we have a lot of humidity here. Plants grow really well in around 70, 80% humidity. But on the space station, it's around 40. It's more like an office building where you're dehumidifying, because they don't want a lot of mildew growing on the walls and things like grow outdoors in Florida. So not all the plants are very happy under that low humidity. And then there's carbon dioxide. So the carbon dioxide on space station, it's around 3,000 parts per million, but it varies depending on how many crew members you might have there. Our carbon dioxide on Earth right now is around 450 parts per million. And there's a lot of research that shows as you increase carbon dioxide, plants tend to grow faster and bigger to a certain point. When you get to too high a level, some plants will actually shut down a lot of their processes because this becomes a stress. And that level may vary depending on different plant varieties. So when we grew the Chinese cabbage on space station, that plant variety was not happy. And we've done a lot of research that traced that directly to the high CO2. That variety did not do well under high CO2, especially when combined with something like an LED light. Okay, when you have these LED lights, it's kind of narrow wavelengths of light, and that's something else that plants haven't evolved to deal with. Real intense light in very narrow bands is not something that they see. They see the sun, which is broad spectrum. It's very intense, but it's got a whole bunch of different wavelengths in it. Whereas with our LEDs, we've just got a very few. And so that can cause some physiological plant disorders. We see some things in the peppers that we grow that are also in response to light. When you have light without UV, certain types of plants will form different problems on their leaves, things that look a little bit like tumors or expanded epidermal cells. So all of these things have to be tested. And so we test different varieties of plants to see which will do well in our environment. But we're also looking at, can plants be engineered to do well in the space environment? And I think there's a lot of potential there. This isn't something that people have looked at very much. Many types of crop engineering have been for the field, for growing outdoors. So when we're talking about growing plants indoors, we want plants to be very small. We want them to be very productive. We want them to be resistant to some of these stresses like overwatering and underwatering and not getting enough oxygen in your roots. And ideally for space crops, we would like them to really help enhance specific nutrients that might break down in the packaged diet. So we might want, a lot of vitamin K or a lot of lutein, which is really good for your eye health, because eye health is important for astronauts. There are some vision changes that go on in space. so these types of things could be specific targets for taking a crop variety that already exists and turning on or off specific genes to help really enhance that to grow well in space, as compared to growing here. So I think we're not doing that right now, but we're certainly open and interested in work of that type because, you can only do so much to get the environment right for the plant. So maybe we should be doing an equal amount of getting the plants right for the environment.
Wow, that's an interesting perspective.
Mm-hmm.
That's a good point. Oh, come on, teachers. I know you've got some questions for us, right?
I have some questions from my students, Dr. Massa. And
Great.
I was hoping that you could talk a little bit about your background and how you got involved in plants up in space and your whole education piece. I think it's very powerful for our students to hear.
Oh, I'm very glad to. So I was, when I was a kid, I grew up about an hour and a half away from where I'm at now, and in Central Florida. And I was fortunate enough to be able to see, the space shuttle launch from outside my house. So that was really exciting. And my other interests included horses. I loved horses. So I decided to take an agriculture class when I was in 7th grade. I was really interested in horses, but also in plants and food in general, as most of us are. And I had an agriculture teacher who was just wonderful. He really inspired me. And he was invited to Kennedy Space Center to learn about the research that was going on. There were a number of teachers that were invited. And so he brought back hours and hours of video and information about the work going on to grow plants for the astronauts. So I still liked horses, but I found that I was really good at horticulture and I really enjoyed learning a lot about plants. And I decided when I saw all his videos that was really what I wanted to do, that I wanted to be able to grow food in space. And so he helped me with a science fair project. We were doing some hydroponic production of tomatoes based on the things that he had learned. Then when I was at high school, my agriculture teacher there also gave me opportunities to build some hydroponic systems for different crops and to grow plants in a greenhouse that we had just sitting on the property that nobody was using. So I got a lot of practical hands-on experience growing plants, and I had some problems. I had a lot of problems. I got this weird fungus in the roots. So I wrote a letter to Kennedy Space Center when I was in high school saying, hey, I've got this problems with my plants and, I'm trying to do hydroponics and I want to learn about plants in space. And I got the most wonderful letter back from my current colleague, Dr. Ray Wheeler, who has been here for a long time, and he's just one of the most outstanding figures in the field of space plants and life support systems. But he took the time to write to this high school student, and he sent me a really nice letter and all of these articles about the science that was going on there, and that was really so inspiring and powerful for me. And now I get to work with them, which is amazing. But, it wasn't a quick journey. I went to undergraduate school. I went to Cornell University, and I studied plant science there, and I got to do more research in the lab. And then I went to graduate school at Penn State. I got to work with Dr. Simon Gilroy, who's the advisor of Richard Barker, whom you met. And so he works a lot more on the fundamental, plant responses to the environment. So I was able to really learn how plants sense and respond to gravity and to mechanical stimuli through my PhD with Simon. And that was a great experience. But I realized then that I was missing the agriculture side. I really liked the biology, but I really wanted to get back in touch with the agriculture. So when I got my PhD, I did a postdoctoral research fellowship with Dr. Carrie Mitchell at Purdue University, who was directing a NASA center for crops.
Any
There a question?
challenge or conflicts, anything with like the meetings that, the chat times, anything that conflicts with or concerns, conflicts with anything or any concerns you've had this week
Can
now that
you
we've kind of
open
got
up your mic,
these
please?
firm times.
I think somebody may have an open mic. Okay. So anyway, I was able there at Purdue to get more research on crop plants and horticulture and build a lot more hydroponic systems and grow crops that I could eat, which for me is really nice. That's one of the things I love to do, and be able to eat your research. And from Purdue, I was able to come here to Kennedy Space Center, and I got hired on by NASA. So it was a long journey, but it was also a really wonderful journey because I met so many great people along the way, and I had these wonderful mentors who really helped me to fulfill my dreams.
That's such an awesome story. I just want to give a big shout out to all the teachers, those that are joining us here, those that are now working in a completely insane environment of your students at home. And you really are an inspiration for the future. And Dr. Massa, you're a perfect example of someone in the field taking the time, being passionate about what they do and sharing that. So I just want to give a big thanks out to all the teachers. Your work.
I agree.
Sometimes we don't see it right away. But I like to say, we don't necessarily look for a return on investment. We look for a return on education, right? And teachers make a difference. And it's really amazing when they take the time, because look what happens. It's so cool.
It is, definitely. I agree with you 100%. The teachers are the best. So.
I have a question. My name's Lori. I'm from Tahoe in California, and I teach elementary kids at 2 schools. So I have about 700 kids, and they've had virtual tours of the space station so they understand the space constraints. So when we talk about plant projects, they're interested in how do you keep plants in a contained environment so they don't just take over the space station.
Yeah, that's a really good question. If I had my way, I would have them take over the space station. But obviously, there's a lot of other important science that's going on up there. And so, veggie, veggie has these walls, these bellows. And actually, it's kind of interesting because when you think about, people were asking when we had problems with the fans, just open the bellows. But that actually won't solve the problem. That would actually make the problem worse, because without natural convection up there, when you don't have fans, you don't have the airflow to bring the gases to the leaves, to bring carbon dioxide, to take away oxygen. You don't remove your water vapor. And so when your bellows are all the way up and attached, so we call that, closed, they're all the way up, that's when the airflow actually works well, because we have a fan that will then draw the air through the plant canopy. But if you have your bellows down and so they're open, the airflow will just bypass the plant. And so really a lot of it's how you design your hardware. If you're putting light in one location, the plants will grow towards that light. If you have airflow, that airflow is going to be very important for, keeping your plants healthy and keeping them from getting fungus or disease. And then you add water to certain regions, and that's where your roots are. And the roots are contained in our plant pillows right now. We have another piece of hardware up there, our advanced plant habitat, and that's a bigger chamber, but it actually doesn't have that much bigger growing area. That one's more controlled. So it will control, has sensors for all the different aspects of the environment, and it can control the environment. But again, the roots are contained in a tray. And so a lot of it's just really about containment and partitioning and being able to to keep the plants where they're going to grow well. So we also have to choose varieties that don't get too out of control. One plant I quite like growing is sweet potato. So sweet potato is a really fun plant to grow. It's got these big vines that'll just go everywhere. It grows like a jungle, and then the roots are where you get the sweet potatoes from. But you can actually eat the vines for certain varieties. You can eat the leaves. But that one, you have to keep the vines in place. Now, we haven't grown it yet in space, but we likely will someday when we have an ability to cook in space. And so we did an experiment showing that you could take these vines that get really long and you could just coil them around a frame or a structure, or just clip them, to a wall to keep them small and compact. So a lot of it is also what we call horticultural technique. How do you maintain your plants to keep them in that growing area? Things like pruning could be done, where you just, like you prune your garden plants to make them smaller and compact. But
kind
a
of
lot is picking varieties. Mm-hmm.
So kind
Go
of
ahead.
the idea of a bonsai, you're trying to train your plant to live in the smaller environment in some respects, and then picking varieties that aren't going to get huge.
In some respects, yeah, but the big difference between bonsai and what we do is bonsai plants are not productive, okay? You don't want a bonsai tree to be really, really productive and to produce a lot. You want it to kind of stay small and compact, and so you nudge it to do that. We kind of want the opposite. We want productive, fast-growing plants, but we have to make sure that we harvest them, often enough, just to keep them contained. So one of the things we do is called cut and come again, where we go back to the same plant and we just cut off the older outer big leaves, and then allow the little leaves to keep growing. And so this works really well for a lot of varieties of leafy green crops like lettuce or Mizuna mustard or Chinese cabbage, because then you have a kind of a continuous source of food. And we've talked to some of the astronauts and they say this is great also because it makes it easy to access the plants, to water them, to make sure there's no problems going on. If you get a really thick, dense canopy, then you don't have that easy access and you break a lot of leaves when you're trying to get to water them.
Awesome, thank you.
A good question.
Hi, Dr. Massa.
Any other?
I'm
Hi.
Diane Phillips. I'm from Canada, Montreal, Quebec. What an amazing presentation this is. Thank you so much. I feel like I'm a kid back in school again, but I love it. One of the things that I'm really curious about, and there's two things. One is the plant pillows. So I want to really hear a little bit more about that. And what is the life like for the plant pillow? What is it made out of? Has it been different in the past to where it is now? What kind of plant pillows, perhaps, I'm going to pose that to the students. For example, what could they come up with for plant pillows? We typically use Ziploc bags, but we'd like to get away from that. So I'm very curious about that. And then the other question is, I work a lot with Let's Talk Science, McGill University, and CSA's Tomato Sphere program. And so we get a chance to do all this regrowth, and they do this in the States as well. What other plants or are we ever going to see any other seeds coming back so that we have that opportunity to maybe grow some of the seeds that are being harvested and returned to Earth?
Yeah, those are great questions. So first, the plant pillows. So the plant pillows are, they're made right now out of a material, it's Teflon coated Kevlar. Okay, so Kevlar is the stuff in bulletproof vests, right? But Teflon keeps things clean. And then they actually have fabric on the bottom. It's a Nomex fabric. Now, a lot of the materials that we select have to be selected for space because they're fireproof or flame retardant. So they have a lot of characteristics that we probably don't need for our classrooms. I make some plant pillow analogs, so things that I can use in my ground testing, and I get like a silver metallic static shield bag, the ones they put electronics in, And then I will just sort of heat seal a little fabric in the surface of that to make a plant pillow that works really well on the ground, and it's a lot cheaper than these hand sewn ones with Teflon and Kevlar. The plant pillows have a little bit of a tubing inside. It's a ring, and it's got little holes all the way around it, and that's how the astronauts add water. So we fill the pillows up with this ceramic, this baked clay, which is the same stuff that's on baseball fields between the bases. It's a sports field amendment called turfus. And then we fill them up, and this ring sort of sits in the middle of the clay. And the ring has, a straw to the outside, and that's where the astronauts add the water. They use a little quick disconnect to add the water from a syringe so that water doesn't leak out. And that quick disconnect is kind of one way. So only when it's open can water go in. And then, we have a one-way valve on the syringe. So we're only putting water in, we're not pulling water out. And we also have a controlled release fertilizer in this clay that slowly releases nutrients over time called NutriCoat. And we've worked with the fertilizer company that they help us figure out which formulation will work well for which type of plant. So it's kind of heavy, the clay and the fertilizer, all this is heavy. So that's one other reason it's really not sustainable. So for other substrates, we're looking for things like foam that might work, things that'll be lighter but could still provide the same sort of water and air containment. The clay is porous, and because it's porous, it helps to trap water and air in the root zone, but it's still not perfect. And right now we can reuse these pillows, but you have to take all this clay and roots and stuff out and wash them and then reuse them. So it's pretty arduous, and it's not something you can easily do in space. So we're definitely interested in new approaches, and, the only things you have to remember are containment. You've got to be able to contain whatever inside, and then trying to provide the nutrients, the water, and the oxygen. You don't need a lot of structural support in space because, there's no gravity, right? And so things aren't going to fall over. But on the ground, to be able to test on the ground, the roots have to have something to hold on to structurally support the plant against gravity. So then, okay, that was that one. Oh, man, I've already forgotten. What was your other question again?
Well, Dr.
So
Massa.
the other question was about seeds that are returned to Earth for mass growth through school,
Right.
and so on, and
Tomatosphere.
have the information about whether they're doing that with anything else in the future.
Yes, sorry, my attention span is short. Yes, no, Tomatosphere is a wonderful program, and we've been really excited about that. Right now, we don't have any other plans to return other seeds. We did get some seeds back from the zinnias, and we were able to grow second generation zinnia seeds from those. But we didn't get that many seeds back from those that were viable. Most of them weren't viable. We didn't really have the astronauts actively pollinating the zinnias or anything. We are going to be growing the tomatoes and peppers, and so hopefully we'll get a few seeds back from that, but I don't think we're going to get the quantity to be able to really distribute them like with Tomatosphere. Tomatosphere, they're just sending up large numbers of seeds, and so then those seeds can be distributed, but they're not actually growing the plants that produce those seeds. So since we can only grow like 6 plants at a time in veggie, it makes it hard to get the numbers of seeds that we would need to be able to do a bigger program like that. I know the UK did a program called Rocket Science, and they sent up arugula seeds to space. And I don't know if there's a way for other schools to participate in that, but that might be something to look at. I believe the European Space Agency has also done a lot more with seed studies. So you might look into those as well and see if there might be an opportunity. I know when Thomas Pesquet was up there, he did lentil, mustard, and radish seed germination. And I think that was associated with schools. So there may be some opportunities there as well, but I'm not aware of any other ones that are coming up anytime soon. Sorry.
Well, Diana, actually, if we want to send some seeds up, we might want to do that. And actually, just on one of our payloads, find a little way to swirl some sweet seeds along. You actually mentioned lentils. That's kind of interesting. Michael Wilkinson, if you can, describe, we're getting ready to do our third set of trials based on looking at a bacteria and its relationship, its symbiotic relationship with legumes. And we're really thinking about something more natural, green fertilizer, and its use off-world. And right now you're talking about like Nomex and Kevlar and Teflon, clearly not really organic material. All those little microorganisms and what have you. What are some of your thoughts around that? Michael, maybe you want to provide an introduction on what this actually is and what's happening there.
Sure. So the Leguminot Challenge, we're really, as Ted said, we're looking at the sustainability of cropping on a Martian or lunar colony, where having the NutriCode or Osmocode or something like that, the up mass is prohibitive. The energy required to produce it in situ and those resources is prohibitive. Using anhydrous ammonia is medically impossible, dangerous. So we're left with a biological challenge. So we're looking to see, will the rhizobia nodulate with the legumes in microgravity and the high carbon dioxide levels and all of the stressors that you detailed earlier in your presentation. We flew cowpeas, Vigna unguiculata, this past flight with their symbiont Rhizobium leguminosarum. And we're presently campaigning, and I see several of our team leaders are on here, campaigning a whole collection of leguminots as our next potential candidates to see if either we're going to refly the same experiment, but the question is, are we going to fly the same plant? And we had some of the same issues that you addressed. I mean, we had fungal issues, we had issues with humidity, just everything. And I've got, we should talk sometime about some theories I'm kicking around in my head in terms of the non-density convection issues in terms of our lab. It's I think Ted mentioned, it's a 2U lab. So a 10 by 10 by 20 centimeter closed. So, we've got added heat issues and added carbon dioxide issues and all the surface tension issues. And it's just like, so there's lots there. So this is really exciting to hear about the work you're doing. So I'll run around the bend. So Mary is sponsoring mung beans. Diana is sponsoring soybeans. Lauren is sponsoring chickpeas. Gil and Tony are azuki. I'm looking who else is here. Steve is doing lentils. I think Mike was going to join somebody else with peas. I'm championing clover. And then we've also got alfalfa. So those are the species that we're looking at. There may be something I left out, but you get the idea.
Yeah, that sounds amazing. That's really exciting. One of the people you might get in touch with is Dr. Gary Studi. He had a payload on the space shuttle. Well, it was on the space station called Synergy, and that was
with
using...
Mike Roberts, right?
Yeah.
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 They did soybean seedlings and short term. I've had the opportunity to talk with Mike a little bit, but I've not been able to get in touch with Gary.
Okay, yeah, I can get you or get Mary's contact info, and he, it was Medicago. They were using the alfalfa, the Medicago, for one of their experiments. They've done a couple, and then the Cynorhizobium. I think definitely there's a tremendous potential for mycorrhizae, for nitrogen fixating microorganisms, and then we also haven't even really talked about the other big sustainability factor of taking the inedible plant material and all your wastes and turning those into resources to recapture all the nutrients that you're going to need.
Yeah, exactly.
I think that's critical.
I mean, Don
calculus
Pettit,
is one of my
Don Pettit, bless his
favorite
heart,
classes.
with all his experiment. He did to a little on a micro scale with his Astra
Yeah.
zucchini. Did some composting.
Yes, yeah, his compost tea that he made for the zucchini. I know
Right.
that was such a wonderful blog. If people haven't
It
read
was.
The Diary of a Space Zucchini, definitely look that up. It's great.
Yeah, and if you're on Twitter,
Um,
at Astra zucchini.
Awesome. That's cool. I need to follow that.
Well, she's not posting anymore, but
Oh, that's a shame. Yeah.
Yeah.
That's so great. Yeah. No,
about,
I think.
the media. I mean, you're using Arcelite,
Yeah. Mhm.
and then, you made me think. Has anybody? You were talking about mass, and I'm always thinking about mass. Is aerogel a candidate for a growth media? Has anybody experimented with that?
Yeah. Aerogel.
Those who don't know,
is something
aerogel is
that
like there's
people are
almost no
interested
mass
in, but
to it.
right. That tends to be kind of closed cell, so I don't know
Okay.
how well the roots
Yeah.
could penetrate. A lot of people have worked on the super absorbent polymers, things like what are inside diapers. But there's been a lot of problems with that in growing plants. We've had a number of people try to test it, and it just does not seem to have super happy plants coming out of it. But we are Trent Smith, who sadly couldn't be on today, but he's the veggie project manager. He's actually a polymer chemist by training, and he's done a lot of work with aerosols and other things. So he's very interested in looking at polymers. But again, it's something, that you'd have to be bringing from Earth. And so ideally, it would be great to either be able to regenerate a substrate. If you need a substrate, maybe make something from your inedible plant residue to grow your plants, whether it's some sort of 3D printed cellulosic sort of thing, or to just not have to have a substrate at all. So, but yeah, that's where we need a lot of great ideas. So.
We're going to have to try to get Trent back on. Our plan here with the uncertain, I'm here in California, and it's unlikely we're going to see a regular school coming back in. And so we're probably going to extend our sessions as we think about design thinking for our future experiments. Exolab 8 is going to fly in October on SpaceX 21. And so this down selection of the legumes is the first step. But we really, I think, what you're opening up in my head is some design ideas, and we're going to work with our teachers that you see here, as well as with our students, to be thinking about some different delivery mechanisms, currently working in with these agar in these tubes, and really not seeing the outcomes we'd like to see. Of course, we're constrained by this 2U equivalent, very small volume. Convection is an issue, and we've done some things to monitor the fans, and we've watched temperatures becoming a little more moderate, but they're still too high. And so in a way, you've kind of, I think all the neurons are firing on the collective brains we have here about what these opportunities are. We encourage you to continue to share your time with us, but we also know how important your time is to you. So we have another guest speaker. I don't know if she's on yet or not, but feel free to just share and continue the conversation with us. But I also am respectful of your time since we're on the top of the hour.
Yeah, unfortunately, I do have another meeting that I have to get to, but I'd be glad to come back and chat more with you, and maybe we can find a time when Trent can join too.
That would be awesome.
Another thing to keep in mind, we had, along with Fairchild Gardens, they did a maker challenge to maker spaces. And so
I saw that. That was so cool. I was talking to Gail yesterday. Yeah.
Okay, great. So partnering with some maker spaces might also be a way to help you guys solve some of the watering challenges too, get some of that, manufacturing brain power involved. I think it's pretty exciting.
Well,
but
it's been such
also
a
your
pleasure.
students, they're so creative. So that's what I love to hear, the student ideas, because usually they're the best.
Well, that's totally what we're hoping to capitalize, is hopefully our students are having opportunity to be bored, which is so critical for creative thinking and insight, and they'll come up with, who knows what. Right. So you have given us so much to think about. I've got to rewatch this hour. I tried to take some notes. It was funny, there's so many of our questions you totally addressed, things that we're trying to think about and things we're questioning. What do we need to think about? So this has been incredibly valuable to my associates here, and I hope the teachers watching this can I know the energy will translate, but really invite your students to run with this and just experiment, play, have fun, and let us know what you learn.
Awesome. Dr. Joy Massa, thank you
Yeah.
so much for sharing some time with
you.
us, brightening our
Thank
day and our
you.
week. We'll reach out to you soon, and hopefully you can join us. Maybe Trent can join us as well in the future. Super
Yeah,
cool.
my pleasure. And I'd love to get some other of my colleagues in and you know, they'd really enjoy this. So
That'd be
thank you.
fabulous.
Yeah, maybe Mr. Wheeler would be kind of cool.
Yeah, we could certainly tie him in. I know he's teaching a class in Canada today remotely. So
Oh, cool.
he loves to do outreach as well. All of our team does. So thank you for the opportunity to speak with you all today. And you have a wonderful day.
You too. Thank you so much.
Thank you.
Thank you.
Thank
Bye,
you.
Dr. Massa.
Bye.
Thank you.
Bye.
And I think Janelle popped in. What's up, girl? You're on mute.
Hello. Hi.
Awesome. Great to see you.
Nice to see you all too.
How you hanging in there? We haven't chatted in a while.
Pretty good. I'm able to basically do my entire job from home. So life has been continuing as normal, but kind of actually feels like more work since
I know, right? I'm here in
yeah.
my garage and it feels like I'm doing more work.
Yeah.
I love that headset.
Oh, thank you.
Why don't we start by talking a little bit about like what you actually do, since you get to do your work from home and you're working at JPL. What kind of privilege is that? Share a little bit what you're up to, and then we'd love to dig into like your story and what have you.
Sure.
And if we can, this live stream, of course, as you know, and we have teachers. I think we have parents that are looking for things for their kids to do. We probably have some students watching us, and you've really thank you so much when you join us sometimes for our sessions live in person. We really know you make a difference. If anything, I think people look up to what you've done and what you've achieved, and there's just so much ahead of you. It's now, but how do you get to be so lucky to be able to work from home and still do the same thing at JPL? So what's up with that?
Well, to be honest, if this happened maybe a decade ago, I would not be able to do my job from home and probably have some kind of essential worker type thing to keep the satellite alive. So my job at JPL is to do instrument operations engineering, which when I got hired, I had no idea what that entailed or what that even meant. But I quickly learned. I joined the Cassini team, and my job on that project, and as some background, Cassini was a spacecraft that was at Saturn, and the mission ended in 2017. So not long after, I actually started at JPL in 2016. And when I joined that project, I was told that it was going to be my job to command the cameras to take images of Saturn, its moons, rings, which is just unreal. The process to do that is it all happens here on the ground. And this is something I like to kind of point out or explain, because a lot of people that I bring to JPL to visit and I'm explaining all the cool things we're doing, it's kind of like, how in the world are we doing all this kind of stuff from here on Earth? Because, there was no astronaut on board the Cassini spacecraft. So who in the world's controlling it and how? And the truth is that every mission is different and the way you operate it is different based on where it's at. And so for Cassini, since Cassini was so far away at Saturn, sending commands to it take around an hour and a half just for them to reach the spacecraft. And that's traveling at the speed of light. And Cassini receives the commands, and then it takes an hour and a half for you to know that they were actually received. And so, as you can imagine, that means you can't really joystick satellites that are that far away, because you would have no idea if your input actually got there or if you're doing the right thing. So instead, we have teams of really smart navigational engineers who are able to predict where we will be over a long period of time and with really great precision. And that allows us to plan ahead because we know where we'll be, we know what we want to see from the scientists who input their needs. And then someone like me, an operations engineer, uses software on the ground to actually generate the commands to get the job done. And we send them off to the spacecraft.
Wow, that's super cool. So you're commanding some spacecraft from there?
Yeah. So since Cassini's over, right now I'm commanding a lunar, an instrument on a lunar spacecraft called the Lunar Reconnaissance Orbiter. And I command an instrument that JPL built called the Diviner instrument. And Diviner is a radiometer that's taking the surface temperature of the lunar surface. And so LRO has been in orbit for quite a long time now. And the primary mission was to basically make a map of the moon. And we have finished that. So now we are looking at particular science objects of interest to learn more about where we should send astronauts to make a base in the future. And so my instrument is being used temperature wise. We found a correlation between the temperature of what we detect and where areas are very rocky, and also where areas are really cold and may contain ice. And so it's been very useful to getting more information about where we should and shouldn't land based on that kind of stuff.
Where do you think we should go?
I think we should, it's just a hard question. I think we should land somewhere close to the ice cap so that we have access to water, which is really great if you're able to if you're able to get a resource like that in situ or from the body you're at itself and don't have to bring it with you because that's so expensive.
Mhm.
And then also an area that is flat enough that we can confidently send more supplies and equipment and bases in the future.
Well, there you go. Anything close to water would be good for all our plants, right, guys?
Absolutely.
So you have a really interesting journey. Why don't you share for us here how you got to JPL and what inspired you? We heard from Dr. Massa, her background and being inspired and just kind of reaching out to someone that was in their field. But I think you have a really interesting story to share. So why don't you share with us how you got to JPL?
Sure. I think my journey really starts when I was younger. And by younger, I mean when I was a kid. And that's because my dad says, he didn't care if he got a daughter or a son. He just wanted his kid to do what he liked. I still think he wanted a son. And I came first, but that meant that I was out there flying kites, launching rockets in the park, doing sports, playing with bugs, getting into video games. And then I also had the other side that my mom presented to me. I like dolls, but I will say I was launching my Barbie out of windows and seeing which one survived. And what this did for me is that it opened up all the gates of what I could and could like and what I could become curious about. And this was really important for me growing up because no one in my immediate family, was in STEM in any way, shape, or form. My mom has her associate's degree and she's a business administrator. And my dad is works in warehouses, and he went to a 4-year college, but he never finished. And so there was never a grand plan or scheme for me to do great things. My family just was there and supporting me, which is the most important thing they could have ever done. So it helped that I didn't have some kind of pressure looming over me and could just generally explore what I wanted to. And so going through school, I really just liked to learn. And I was one of those self driven kids, which I know is not the case for a lot of other students and for various reasons. But I also wasn't just a math and science person. I loved to read, I loved art. Even if I was bad at it sometimes, but I just loved so many things. I just wanted to learn as much as I could, and I really only learned about engineering when during Bring Your Child to Work Day. My mom worked at a company or still works there that had lots and lots of engineers. So when I went to her job, my eyes kind of like got big. I'm like, wow, look at all these people. They're so smart. And look at all the things that they can make. And that's kind of what my understanding of engineering was: people who make stuff, all sorts of stuff. And so in high school, junior year, I was starting to get a whole bunch of letters in the mail from colleges all over the country. And I had not heard of most of them. I'm going to say 90%. If they weren't from New Jersey, where I grew up, and I didn't know about them. And so after a certain point, I just started to kind of throw them in the trash can because I was shoving them under my bed, but I started to lose room. So they were just going in the trash. And on one particular day, I was going through the mail in the kitchen and I got this pamphlet from a school called MIT. Never heard of it in my life. It seemed kind of cool. They talked about something about Guitar Hero, which I was into video games. So I was like, oh, that's nice. But it was in the pile. And as I was walking it over to the trash can, my mom stopped me and said, "What are you doing? MIT is a great school and you're going to apply." I was like, really? Because the pamphlet was actually for a summer program that they offered every year on site at the school, where they invited 70 students from around the nation to come free of charge and stay there for 6 weeks. And it just seemed like a lot more work because I was taking lots of classes. I had never heard of the school before, but it's like, all right, mom, fine, I'll apply, I'll do this thing. So in typical me fashion as a procrastinator, I submitted it the night before, right before the deadline, and went on with my life. And I was a couple weeks go by and one of my classmates asked, "What are you going to do for the summer?" And every summer up to that point, I ran cross country. I was into sports a lot. So I was cross country runner and I did track in the winter and track in the spring. And it's just a cycle year round. I see your thumbs up, Lori. And so that's what I told him. I'm probably going to practice for cross country. I'm like, unless I get into this program that I applied for. And he's like, well, what's the program? And I said, it's for this school called MIT. And he was like, MIT? Do you know what that school is? I was like, ooh, apparently not. So that night I went home and for the first time, I actually Googled the program's main page and looked through it. And that's where I found out that in the previous year, over 2,500 students applied, and one student from New Jersey was selected to go. And at that point, I was like, Mom, you definitely wasted my time. There's just no way, absolutely no way. If I had known that information beforehand, I would not have bothered to try. So I'm at track practice in the springtime, and my mom gives me a call and she says, Janelle, you need to come home. You have a package in the mail. I'm like, well, can't it wait? And she's like, no, it can't wait. It's a package from MIT. I'm like, well, Mom, we went over this. I'm not getting in. She's like, well, you need to come home. So I'm like, fine. I hop in the car, get home. My mom was waiting at the door for me with it in her hand. And she's like, here, open it. I'm like, Mom, stop getting excited. They're just telling me I didn't get in. And she looks at me and she says, Janelle, they don't send you a package to tell you no. I was like, oh, you may have a point there. So I rip it open and I read the first line and it says, congratulations. And oh my gosh, we were jumping up and down, having a celebration. We could not possibly believe that this was happening. This unattainable, unreachable thing. There's no way you can get in. And I was not a perfect student either. I didn't have straight A's. I didn't have a background of people who could guide me to writing good essays. I just applied as myself and MIT accepted me. And that just meant the world, the world. So I end up going to the summer program. This is the summer after my junior year. And I fall in love with this school and the people around me. And for the first time, I realized that there are other students out there who are really invested in their education. I went to a public school and it definitely wasn't the norm to care about your schoolwork. It was not the norm. And a lot of the classes I took a lot of AP classes. My classmates cared, but everybody else kind of didn't. And you kind of were looked at as a geek or a nerd if you did. But for the first time, I was surrounded by all of these students who shared the passion to learn, who were willing to stay up until 2 a.m. just to get the question right for a math problem that didn't even count as a grade. It's not even a real grade. And I just love that environment. So I went from not knowing where I wanted to go to college at all to now saying, I want to go to the school MIT. And so my senior year, I applied early action, and I freaked out every single day after that application went in, especially since right after I submitted the application, I took a look at you could print out what you submitted. And I didn't have anyone look over my application, which big mistake because the first question I had made a typo. And I was like, you know what? That's it. I'm done. I don't want to look at the rest. I messed up. So I panic. And I'm at the bowling alley the day that announcements are supposed to come out. And they at MIT, they come out at very strange times. Like they do like tau time or pi time. So down to the minute. And someone was like, "Hey, Janelle, I think it's your application results are in now." It's like, "Oh my gosh, you're right." So I run into the bathroom stall at the bowling alley. I shut the door. I have my little phone. I'm navigating, trying to get to the page. There's a lady in the stall next to me. And I open it up and I see that word congratulations again. And oh my gosh, it was just the most incredible thing in the world. I had my little party in the bathroom stall at the bowling alley. And it was just amazing. Oh, dare mighty things, the JPL mantra. And so after that, I went to MIT. I still had no plan. I was never the kid with the plan. I just got there and I was like, I really love math. Maybe I'll become a theoretical mathematician. But after talking to some upperclassmen, they were encouraging me to just try an engineering class because MIT is just known for their engineering classes. And so looking at the list, I decided to go with aerospace engineering, mainly because it had the word space in it. And space is pretty cool. So I go to that first introductory class, and our professor is at the board and he's going over our syllabus. So we're going to learn about rockets, we're going to learn about high altitude balloons. He's like, we're even going to learn a little bit about the history of spaceflight. And he pulls up a picture of an astronaut fixing the Hubble telescope. And he says, that was actually me back in the day. I was like, what did he just say? My professor is an astronaut? Everyone else in the room seemed to completely had known this beforehand. They had done their research. But I was one of those few whose mind was just being blown that I, someone who had never had contact with anyone NASA space related in my entire life, now I had an astronaut right in front of me and he was my professor. And this was a game changer. I went from not really knowing what I wanted to do to, I think I'd be kind of crazy to pass up the opportunity to learn about this stuff from a real life astronaut. And so that's the major that I pursued. And at MIT and in the major aerospace engineering, you could not go through a class without someone mentioning this magical place called JPL, the Jet Propulsion Laboratory. And I had never heard of this place before in my life, but we would be in class and we would go over some kind of equation and say, well, let's say we're trying to land a rover on Mars, like JPL, or let's say we're sending a spacecraft to Venus, like JPL. I'm like, this place must be something because everyone is talking about it. And so at our career fair, of course, I'm lining up in the JPL booth. I have my resume ready. My classmates are in line with me. And I remember that first time being in line and overhearing the conversation between one of my classmates and the recruiter. And it basically just made me feel like, oh my gosh, I am not qualified. My classmates were talking about how they were working on labs and projects that were going to be sending something on a rocket in a couple of months, and how they had done some award winning research. And I'm looking at myself like, I don't have any of that. So that was wake up call number one. I need to do something other than just my schoolwork. It's not enough to just exist, go through the motions, and be in school. You need to find what you're passionate about and then continue to do that kind of work outside of the classroom. And so that was I found that easy enough to do. I was very passionate about outreach work, and I had joined a club for that my freshman year. So I was going to continue that work. And then I was going to start up a new outreach branch of the National Society of Black Engineers chapter at my school, where instead of just us raising money for us to go to conferences and things, we would do outreach to the local high school that was a junior chapter for our branch. And so what I ended up coming up with was launching high altitude balloons and having them send experiments on them. And it was such a fun time to spend with the kids and to also run something like that entirely on my own based off of all the things I was learning in my classes. It's something that I could realistically pull off. And so I was also starting to feel more like a competent engineer and not just a student struggling to stay afloat. And I would like to say that I improved myself every year at MIT, but I still applied that sophomore year, didn't hear back. I still applied junior year and didn't hear back. I did, of course, pursue other things and didn't put all my eggs in one basket, but the prize was this place, NASA JPL. And so my senior year was my last chance, and I didn't have an internship from them under my belt. And at that point in time, JPL really was only hiring interns who had previously come to the job because those were people that were known quantities. They weren't question marks. But it wasn't going to stop me from applying, of course. I learned to apply to everything after I got accepted into that MIT summer program. And so I'm there, and for the first time, I actually hear back from JPL. And thank goodness it wasn't a no. It was an interview with 3 hiring supervisors who were interested in what I was doing. And I went through the interview, very nervous, of course. And I heard back a couple weeks later that they wanted to fly me out for an on-site interview. And that's when I first got to see the campus of JPL, and it's when I knew this has to be the place. And so after I got that interview over and came back and was worrying and waiting, right before Thanksgiving, I get a call asking if somebody had discussed salary with me yet. And I said, "Salary? Are you saying I got the job?" And they're like, "Oh, no one told you?" Yes, you did. and, and that was the moment I had been waiting for. And that was kind of my journey to where I am now. All sort of just taking opportunities as they appeared and going forth without a plan.
Oh, now you had a little bit of a plan, I think. You were following your passion, right?
Yes, that's true.
And you were, and I think you kept your head down. You were doing your work too. It sounds like if you're studying your AP classes.
hmm
Mhm.
Absolutely.
And something that I think is really important is having that infrastructure of support.
Yes.
Now, we're kind of in a really pretty unusual time for the planet, right? Here we are pretty much all at home in our respective places. We're trying to make as normal as possible. And we have many teachers here with us online directly inside Zoom, and some that are joining us via Facebook and YouTube. And their students are out there, and some students don't even have access to a computer, let alone the internet.
Mm.
So how do we create and reach and create those infrastructures of an inspiration, right? And I think we're seeing here the amazing work that our teachers do, even in the most difficult times. They're stepping up and they're finding a way to help kind of build that infrastructure. Can you recall any of your teachers kind of going through school, K through 12, that really inspired you?
Oh, yeah. I loved my teachers. I loved my teachers. Oh my goodness. I don't know. There's not one that I could just zero in on because there're just so many that I really appreciated. In fact, when I go back to my hometown in New Jersey, my parents don't even live there anymore. But when I go back to my elementary school with my friends, and we go and hang out and say hi. And it's I've always had a very thankful relationship with my teachers because I just saw what they were up against. As somebody who had an innate joy for learning
Mhm.
and who tried to help other students in the class, because one, it just seemed like the right thing to do. And two, it helped me know that I understand what I'm doing too. Um, it just showed how difficult it is. And I used to do tutoring as well, and I would get students, especially for math, who would come in who just didn't who couldn't grasp concepts, and I would find, crazy things. For instance, best way to explain this was a girl who was learning about algebra, and there was a problem that said something like 5x equals 50. And so we were trying to figure out what X is. And she was saying, well, X is 0. And I put two and two together to realize that she had missed a crucial step of what the X actually is and what it stands for. She wasn't multiplying, she was making 50 by adding the 0 to the 5. And as soon as we got through that, she understood. But when you have a class of 30 students and a bunch of them could be having misconceptions, different misconceptions, it's like, how do you even have the time to sift through and figure out where the disconnect is happening? I don't think it's as much that students can't do it, is that if they miss the ball, especially early on, and they can't figure out where that disconnect is, then they start to become unmotivated because they don't understand what's going on anymore. And for math, since you're constantly building on what you've learned before, if you miss something very early on, you could feel like you don't understand this thing. Whereas people who really love math are like, math is great because there's always an answer, there's always clear steps to take, there's instructions, you can't fail. So is I really have a lot of respect for my math teachers who have to deal with that.
Yeah, so X doesn't equal 0. I think I get it now.
Right. Right.
Well, also, I guess, oh, there you go. Look, here's Janelle with us.
Oh
And
my
this was
God.
down in Garvey, east of LA. It's quite an inspiration. We launched a balloon that day. That was really quite fun. And what we got, there you are. And the crew you hang out with is super important too, right?
Mm,
I mean, you get inspired not just from your teachers and support from your family, but it's your friends. And you have
absolutely.
friends there's good kinds of trouble and there's bad kinds of trouble, right?
Yes.
And so getting into situations that may not make a little uncertain, uh, having the right crew around is pretty important too, wouldn't you say?
Yeah. And I was lucky that the friend group I made in elementary school has followed me all the way to this day.
Mm,
And so they are people who I could I could bet my life on them. I can always trust and rely on them to always have my best interests in heart.
mm,
But my brother had a very different experience growing up. My mom was always so confused because she always thought that girls would be the ones who bicker and get in fights all the time. But no, it was my brother and his friends were always the ones who were getting in fights, getting mad at each other, getting into trouble. And as time went on, then though, my brother started to learn to cut people off who were toxic, and it was
mm,
definitely hard at first,
mm.
definitely hard. He would hang out with a boy spit on him once, and they were back hanging out the next week. It's like he finally started to understand that, choose friends who respect you and who you can rely on.
Mm,
And once he started doing that, the fighting and the bickering and stuff really started to die down. But high school is just it's is such a high school and middle school, and it's middle school too, honestly. They're just such toxic environments. I think that sometimes we forget how absolutely ruthless kids can be to each other.
mm,
And the fact that you have to do schoolwork on top of deal with the social implications and not fitting in, it's absolutely crazy. Popularity, it's still a thing when you become an adult, but it's not as much of a thing. It's more like, are you famous or not? And everyone else is kind of equals.
mhm,
But in school, there is such a tiered system that can make you feel like even if you're interested in something, you can't show it outwardly. And so I've learned that, you know, how can I do my part in trying to help these students, especially students who came from backgrounds like me, who had very little guidance, but still want to do something amazing. And that's just speaking with them, letting them know that I am a human being. I'm nothing special. I am not a genius. I didn't just get born. I was not born with smarts. I was teased when I was in school. I remember kids saying that I looked like Oprah when I was in middle school because I had like a bob cut. And that was so insulting. I was like 10, 11, 12 years old being called Oprah. It's not, and it just, I understand what they're going through and how hard it may be to outwardly show that they care about something. So it's not so much about the instant gratification of a kid running up and saying, "Oh, I want to be just like you." It's more about just sharing the information so that when they go home and are in a safe space, they can delve deeper into it themselves, because you really don't know how they feel on the surface, especially when they're with their friends.
There you go. So with some of our teachers that are there, or anyone have any thoughts or questions for Janelle? I should check our Facebook.
I have a
too.
question.
Yeah. What's up, Liam?
Hey, Janelle.
Hey.
So great to have you on here, inspiring the world with your extraordinary story. I have a question about what it's like for you to now work at JPL. There's two parts of it. There's actually what it was like going to work regularly. For those, so and now obviously you're in a completely different way of working. And I think a lot of people are on here, the teachers and others, sort of like there's a way that now the day is conducted. And they're thinking, a lot of us are thinking about how to educate students, whereas you have a job to do at JPL. And I'm curious of just what things were like before. So like just walking onto JPL campus and getting to do the things that you do to do what you do, and then what it's like now. So that just sort of came up for me as sort of like an interesting day in the life. And over to you. If you can find a question out of what I asked.
Sure. So I pointed out in the beginning that life hasn't changed too much for me, and that's mainly because almost everything I do in my daily life happens in front of a computer screen. Computers are the way that we communicate with the world at JPL. So when I went into work, though, there are a couple of key things that I'm missing now. And the first is that sense of amazement and excitement of seeing the JPL campus. And that's because throughout our campus, we have lots of artistic uh, models and ways to show the public what we are doing. And that also inspired me to walk in, go through a building, and see a full-size model of the Curiosity rover sitting beside me. That is something that, is a reminder, a constant reminder that we are doing amazing things every day here. And I'm just thankful. So it's sad to not be able to walk into work and see all of those things. But normally my day would go, I have two projects that I work on. The first one is the Lunar Reconnaissance Orbiter that I spoke about earlier. And I would come in, I would make sure, firstly, that all the commanding that was scheduled to happen the previous day actually did happen by creating reports and checking the housekeeping telemetry, we call it, which is basically data that tells us the health and safety of the instrument. Are we performing the way we should be? Is it good? Did we get all the data that we were supposed to get? That kind of stuff. Once everything is good on that end, if we don't have any problems, then I move on to make the commands for the next day. And I use software and I use a lot of Linux systems. So I'm often in terminal with the black background and green, just because I like those colors. But we're not hacking or doing anything like that. Just using a lot of remote servers. And so that's the way you do it. And so I'm often in that environment, creating the commands, sending them over to our spacecraft host, which was the spacecraft made by Goddard. And so we send those commands securely to Goddard, and we get a confirmation that they received them. They let us know what they're planning to upload the day before, and we kind of do is this what you sent me? Is this good? And we're like, yep, everything's good. We're ready to go. And a couple of years ago, when I first started at JPL, when we would upload things during the daytime, because we upload in the day and night, during the daytime, I would actually get on a headset like this, be on the phone with Goddard, and one by one for each command, they would ask if the command was okay to send. And I would say, what would we say? It was some weird talk. It was like they would say, oh, we would say, go, it's good for uplink. We would repeat the name of the command, say it's good for uplink, and they would say on the way, and they would hit the send button, and I could see it going up.
Wow.
Yeah, but we ended up actually automating that process with scripts. I'm really big into automating anything that takes more time than it needs to, so I do that really often. So it's really cool to do that each day. You kind of feel like, oh, I'm an operator. Oh, I'm on the phone, blah, blah, blah. Something. Right. But now we kind of set it up so that we have a program that's listening and watching during the times we specify for the commands to go up, and that will kind of yell at you if they don't. So you don't have to do it like that anymore. And so the commands are sent, you check the commands from yesterday, you're pretty much good to go. So for that project, I spend my extra time creating scripts to make our job easier. Like every week we have to update what is on board the instrument and update it into us so that everybody, the scientists know, the science planners know. And it used to be very manual. I'd have to go through our reports and say, okay, we sent this table on this day, so this is the most recent, go through all the reports. But then I wrote a Python script that kind of just looks through everything automatically and updates the wiki page, and you're done. So I spend the rest of my time doing that. The other half of my day is spent on a project called the Multi-Angle Imager for Aerosols. And this project is absolutely amazing and so, so important. So JPL, even though we get a lot of flash from our Mars missions and interplanetary and stuff like that, we also work on Earth. A third of all the work we do at JPL is for our own planet, which is so important. We matter too.
I'd say so.
Yes. And so the Maya mission is an instrument that's going to be on a spacecraft that's going to be observing the Earth to measure pollution in cities globally. And we're not just going to measure the pollution, we're also going to get the health data from those cities to find out correlations between how certain pollutants cause health problems for people. And the reason why this is a different mission is because one, it's the first NASA mission ever to include the science return, like doing that epidemiology, that disease study, that's included in the mission. So one of our requirements is to do the study. So if we don't do the study, it's a failure. This is important. That means you have the money to support the scientists after the fact. They don't have to go running around for their own grants to find ways to use the data. The second part is that everyone knows that pollution is bad already, right? So it's not necessarily that we need to learn, we need to find out that, oh, pollution exists, there's problems with people. That's a known fact. But this instrument is going to be figuring out the differences in the kinds of pollution. And we do it by, we call them particular matter, and it's measured by size. And that's kind of how you can tell what kinds of pollution, what kinds of pollutants are present. And when you can be more pointed and say companies that are producing X, Y, and Z are causing these diseases in people, now you can put a little bit more pressure on them. And so I'm so excited for this to launch. It's projected to launch in 2022. And my job on that project is to figure out how in the world we're going to operate this thing once it gets to space. So it's very much like my other job, except for now that I have the experience, I can have the input. And it's a great experience. And well, this summer, I'm right now, I'm basically in charge of figuring out how we're going to do this on the ground, what kind of systems we're going to use. And that also means that I'm responsible for getting them all tested. And since I'm going to be the operator come time for us to launch, I'm going to be the one in the lab testing all our software to make sure it actually works as intended. And so that's one part of my job that has to be put on hold until we get back to lab. And right now we're in the integration and testing phase. We were calibrating our camera, and then we were going to move on to getting into the lab and putting everything together and start testing it with our software. And so all of that has to kind of be put on pause. But what I'm doing instead is writing up procedures so that when you get in the room, you know exactly what to do. Every step is signed off and it's very organized. And I'm also figuring out the procedures for what are the differences between doing it in the testing environment and doing it when it's in space.
Well, that's fascinating. I'm going to have to take this offline, but with some of our ground studies, that like the CanSat that we launched in the balloon with you down in Garvey, we were looking at air quality, and we worked here in the local Bay Area region with the Bay Area Air Quality Management District on gases, carbon monoxide and sulfur dioxide and what have you, and also the particulates.
Yeah.
Understand that now that I know a little bit about your Maya mission in 2022, I'm going to now try to triangulate and meet you there and find
Yeah.
ways that our students can study their local air quality. Probably through
Absolutely.
more conventional instruments, conventional off-the-shelf components, not necessarily really high-end stuff.
Yeah, that's
That would be
fine.
really fun to kind of make that connection. Not to derail it, but that would be really, really cool.
That's definitely a possibility. We have a part of our team
Mhm.
is focused on finding early adopters,
Mhm.
scientists outside of the people who are funded specifically by the project
Mhm.
who want to learn more about it and want to
Mhm.
contribute to the science. So
Oh, yeah.
absolutely.
Yeah. Did you know also that we're working on putting a digital nose on the space station?
Oh, what?
There's a robot, a series of robots that they're called Astro Bees. They're
Oh,
30 centimeter cube robots, and they have little fans. They're flying around the station. They're just now being set up and tested. But they're essentially operational. But we've got a guest science opportunity to add sensors to measure gases.
oh wow.
Like things like particulates and what have you may be less likely on the International Space Station, but the other gases and gas behavior is different. We were learning from Dr. Joy Massa earlier about gas and water behaviors in microgravity. So that's just kind of interesting connection as well. As we think
Yeah.
about air quality outside, also air quality indoors. In classroom, what have you, if you have a poorly ventilated classroom, students may not be doing as well, right?
Oh.
Oh, looks like we lost Ted
That's
for
my
a second.
internet.
Oh, oh no, there you are.
Yeah. So, yeah, there, I'm back. Yeah, I have a funny thing here, just trying to make the home office work. I got wireless, but when I plug in my wire, I don't have a way to plug in power, so my battery goes down, so I have to switch over.
Oh,
And so
got
I'm
it.
on wireless. Yeah. And then when my battery back up to a level, I'll switch back over to the wired.
Got it.
Anyway, do we
Mhm.
have any questions or thoughts from our teachers that are out there? I see Sam and Lori and that joined us. Great that you guys are on. Cool. Well, we've got,
Hey, because no one else jumped in, I've got a follow-on question.
I'm sure you have quite a few, Liam.
Anyway, so Janelle, yeah, I was really intrigued. You're obviously going from moon-based spacecraft or moon orbiting spacecraft now to an Earth viewing spacecraft. And that brings us sort of back to obviously the major focus, I think, of where a lot of us are focused right now is on the International Space Station.
Mm.
So so that new thing that you're talking about is an Earth viewing spacecraft. Sorry, it's its own spacecraft. I know,
Yes.
yeah. So and I know that some there are now some JPL experiment platforms that are stationed on the International Space Station that are also
Yes.
Earth viewing, like OCO3, just recently,
Mhm.
and what I wanted to draw a sort of an underline through is just noticing the connection between we're doing biological experiments, plant-based experiments on the space station, and you are looking at it sounds like things related to, is it pollution that might impact agriculture on the ground? Is that part
The
of what is that
the
the outcome of?
The main focus is to measure particulate matter in specific cities where people live so that
Ooh.
we can figure out the health implications.
Yes. Okay. So it's human health.
It's human health.
Human health. Yeah. There you go. Okay. Got it. That's helpful. Anyhow, just what you reminded me is that what a lot of people think about things in space. They're about things in space, but really, it's always about life on Earth in the end.
Yeah.
Sort of, it doesn't matter, where you are, you're observing things that are happening around in the solar system, and obviously a spacecraft orbiting the Earth, you're looking at what's happening on the Earth. So I think I just want to give a shout out for just what you're involved in that is impacting the future of life on the Earth. And thank you for being part of that measure in how human beings, how we as a species are looking to the future. And it's not just looking to the future, it's actually creating ways in which you can impact the future. And part of what you do that with is by analyzing and thinking of the experiments that you can do that will give you the information that would allow you to powerfully make appropriate adjustments to the way that we live on the Earth.
Absolutely.
So, anyhow, just so inspired by your story.
Oh, thank you.
And sort of makes it accessible. Sort of like I just love how you brought in, hey, I don't have to be the cleverest person in math. I'm not that. But look at what no one would know that.
Right.
Just what you're up to, it's just extraordinary. And I know what you got present to is that there's a way you think you are in math, and it's based around all of those little micro failures that you may have experienced. I had a lot of those, sort of like feeling like not the cleverest person around. But what I got from your sharing is that that's not a measure of your capability,
right,
to do extraordinary things. And that's what I find is so inspiring about your story, is that just what you can accomplish when you get that inspiration. And you are that for all of us, for all of the young people around. So yay, Janelle.
oh.
Janelle fan club. Hey, so can we talk about the moon a little bit? I want to get something on the surface of the moon, and we want to grow like plants on Mars, right?
Ooh,
And you're looking
yes.
for water, and well, I guess you're measuring temperature, really.
Mm-hmm.
And I understand that, well, gravity on Earth is also uneven. There's like, you go different parts of the world, there's different gravities. Are you measuring that as well? Or do you kind of get a sense of like, this is the wobbly part of the orbit, or is everything always like nice and smooth, or how does that work?
I want to say yes. I think we have some kind of magnetometer instrument on the Lunar Reconnaissance Orbiter, but to be honest, I don't know much about what the other instruments are doing, because they're not JPL based. But well, we don't have conference calls. Well, at least I'm not invited to them to figure out what other science is happening. But there is a website dedicated to that satellite in particular that lays out what each of the instruments are responsible for doing. And so that would definitely give more information on that front, much better than I can provide.
That's really cool. So you've got a number of instruments running by different groups, and then you have someone operating the primary vehicle itself.
Yes.
And is that also JPL doing?
That's Goddard.
That's
NASA
Goddard. All right. And so Goddard, and then you guys are doing temperature and just temperature, or are you doing other instruments on it?
Just the diviner instrument. Yes,
Okay.
the radiometer.
Really cool. All right. And that map, is that public data, or is that?
Yes, that's the best part. Let me just put it in the
last week we had Dr. Barker talking about data science, really, even though we were getting into microbes and plant biology, and he was talking about data sets. And this is always cool when we know that there is information that's quantifying what you're picking up. And then through that information, sometimes it's just so much data that can you make any sense of it? But I guess if you do enough, orbits, you start compiling a representation of what you're seeing, and you're validating what you think you know about the behavior on that body, in this case, the moon.
Absolutely. And the
Really
thing I
easy.
like about most of the, I don't know why it happens this way, but most of the instruments that I operate, some of the products that we get back are images, and that's really easy to share with people and explain what your job is, besides, like, like a plot that's like, I don't know what to make of this. I just added I added in the comments a link to our instruments website. And actually, can I share my screen to
You
show that?
absolutely can. At the bottom there, you see
Okay.
the share screen.
Oh, let me do just the diviner. Yeah.
Cool.
Okay. So, which one is it? Do you see my mouse?
I see your screen. It says LR Diviner Lunar Radiometer Experiment.
Did it move up and down?
Yep.
Okay. I have two of them open, so I couldn't tell. So if you go here to data, and you go to the data
Posted at UCLA, huh?
archive.
Or it was, that one page is.
I should remember how to do this. Coverage, raw data product derived. Let's see.
So this is like the consumer facing, if you will.
It's for scientists.
Okay.
Yeah, we are responsible for making the data publicly available, not just for scientists, but for anyone. It's available to anyone who wants to use them. So maybe look in here.
Now, NASA, well, I've done a couple of these, the space hackathons. And I'm not sure if this data set was ever a part of that. I usually look at Earth, but this would be kind of fun.
Yes, I'm trying to, of course, I can never figure this stuff out when I need to, but the images are available. Not that, not that.
As you're searching, I'm curious what role machine learning or artificial intelligence might have in data sets, if you know of anyone working on that type of stuff as well.
Machine learning? Oh yeah, that's there in my section. We are responsible for retrieving the data products and also processing them. And so
okay.
I'm involved in the retrieval process on the operational side, but then there's a whole team who has to deal with the science data once it gets back on the ground, and doing things like processing it into all sorts of forms that are useful to the scientists. Let's see if this is, there we go. So there's lots of, but this isn't
that
exactly,
makes a lot of sense.
I know, right? This is not exactly
Well,
what I was
looked like
looking
that was
for.
latitude and longitude. I got that real briefly, but
they have actual photos of the moon, but you probably have to dig, because I just cannot remember these things.
Oh, there you go.
It's gorgeous. Yeah. So there's tons of data on this site. I usually am never really looking on here, but it is made publicly available.
I got some extra time on my hands. I wish I did, actually. I think we're working twice as hard now with a lot of teachers, in their classrooms being remote.
Yes, I can only imagine. But I do have to go now. I do have a meeting happening.
Thank you so much. That was really awesome that you could join us.
Yeah, thank you for inviting me.
If you can pop up.
Thank you, Janelle.
Yeah, no problem.
And if you got some time, we're going to be doing these sessions that we were going to run them through Earth Day, because we're really picking a design challenge. We're going to look at the next flight candidate on mission 8, Exolab 8, that flies in October. But we're probably going to extend it, because it looks like, well, definitely here in California, we may not even be able to go back to school for this school year. And so how do we create experiences that are fun and engaging for teachers that can then repurpose it in their classroom, whether it's K-5 or middle school or high school? And I think even some university students have been interested in the work that we're doing. And so maybe we'll create a little tighter format, maybe just talk about LRO or something, or a day in the life of, which would be kind of neat. But you're, again, quite an inspiration as we find the students that are interested in these kind of projects. Is always nice to get it firsthand from someone that's kind of gone through the gauntlet, made it to the other side, and still smiling about it. So thanks again, Janelle.
Yeah, thank
Thank you
you
so much, Janelle. Jersey girl makes good.
Yeah, Jersey, woo.
Awesome. Thank you for your energy.
Yeah, sure. Goodbye,
well.
everybody.
Take care. See ya.
Janelle.
Bye. Thank you.
And I just see Drew just popped on. Hey, Andrew. And I'm not sure if,
Dan Basso is on too.
yeah. Oh, hey, Dan. I see his name.
Hey,
there.
how are you?
Oh, fantastic. Hey, so in our
hopped on.
brief little intermission here, you know, this is kind of like a telethon. It just kind of keeps on going. It's, I think, a little shorter than what's happening last week. Hey there, Drew. But what we might do, Michael, if you'd like, is do just a quick intermission talking about our mission and the reason why all our teachers are here with ExoLab 8 coming up, and we need help, right?
Yeah.
While we wait for a Twyman to join us, why don't you share with us kind of what we're up to?
So we just concluded mission 7. And with 7, we flew a legume called cowpeas. Some of you probably know it by the name black-eyed peas. And what we were looking at is, can legumes function with their bacteria symbiont, rhizobium, to nodulate and fix nitrogen in the stresses of a microgravity environment? And looking forward to how can we use that to perhaps convert a regolith on the moon or Mars into a viable soil as a growth medium. But even if we're using a hydroponic system, we still have to feed the plants the essential nutrients. And Tony's showing the classroom-based lab that's an analog of what our friends Andrew and Twyman built to go on orbit. And now we're thinking about our next mission, mission 8. And we've launched what we're calling the Leguminaut Challenge. We need to identify what legume is going to be the best candidate to fly in starting in October. Do we fly the cowpeas again, or is there another legume that's better suited to the conditions and the constraints that we have? And that's really where the engineering and design challenge comes. Is there's 2 parts. You got the biological part, like which legume do you want to see go and why? And maybe it's just because I like grandma's black-eyed pea recipe, or I like garden peas, or whatever it is. That could be the reason. It could be that you've done some research with your students on exactly what the needs of those plants are, what the benefits of those plants are, and all of that will be considered as we down select to our final candidate for mission 8.
And tomorrow, Friday, 8 o'clock Pacific, 11 Eastern, we're going to be walking through some of those legume candidates.
Right.
If you want a little more information on that, of course, you can go to Magnitude.io. But we couldn't do anything here connecting to really 100+ schools on our current mission. And we've got schools in Africa and Europe and here in North America, without having someone to be able to get our payload up to the International Space Station. So what we do here, I like to define it as the Kármán line. Everything below the Kármán line is what Magnitude works on. Everything once you get into space, we have to rely on a really special company called Space Tango. And Space Tango, I've got one of their little devices here. This has been to space before. It's a cube lab. It's an aluminum box, 10-centimeter cube. And these are the kind of like, think about it like intermodal transportation for space. Without this system, it'd be difficult, very expensive for us to get things up there. And if we imagine it like a building block, when I say intermodal transportation, imagine like a 40-foot container on the back of a truck or on a rail car on a ship. With that system, you can fill that up, 40-foot container, and send it anywhere in the world for a few $1,000s. Now we've got what's cost a little bit more than a few $1,000s, but we've got these little tiny cubes and variations of this. Maybe like a 2U would be twice this length, or 20 centimeters, or a 3U or longer. Fill it up with some lab experiments, adding fans and power supply and data port. We can then run our experiments. And Space Tango, based in Kentucky, it has enabled us to do that. We're joined by phone with the CEO, Twyman Clements, and I know he has a hard stop at 1/2 the hour, and Drew Diddle, who is our biology lead on the Cube Labs themselves. And so, Twyman, thank you for joining us. I hope you got a good, you connected here, but I'd love you to share a little background. Hey, there you go, of Tango and how you guys got started, the origins, and kind of what you're up to, because you're doing some really cool stuff with Budweiser and with brain organoids, and it's all over the place. And then at 1/2 past, we'll turn it over to Drew, and he'll share more about specifically how it applies to ExoLab and these experiments that we're running with plants. So it's all yours, Twyman.
Yeah, thank you very much. And let me, I'm trying to get in a good spot. Let me see if I can get my video up here working. There we go. All right, there we go. Hello, everybody.
Awesome.
Yeah, so thank you guys. Yeah, Ted and Tony and Mike and Michael. So just to give you, obviously working from home like everybody else, Space Tango got started. I co-founded the company at a graduate school several years ago, and taking it way back, actually, this is really the company really came out of an educational opportunity. Now this was in college, but worked for a lab, an engineering research lab that was one of the first to build satellites that launched on NASA rockets. And so worked on satellites in college, free flying satellites, and then got into doing work on the space station. Really understood that, began to understand that process, and then how can you make that easier? And so then wrote my master's thesis on that, and then co-founded the company outside of there. And of course, it took several years to really get the company going. But what Space Tango's focus is using zero gravity, microgravity, as an enabler for new types of manufacturing. And that's in everything across from semiconductors to layer deposition materials to things in the biologics, including stem cells and organoids and things like that. And so what we're really trying to do is kind of build the supply chain and the equipment to do that. And all of this is equipment all of this manufacturing use cases are those for Earth. This is not about sending stuff and kind of going out to the planets. While I'm interested in that personally, I don't think most of the total addressable market is much better below orbit. So we're staying focused there. And where we kind of cross-section with Magnitude in our education work is obviously in trying to simplify. What we're trying to do is simplify for even the education marketplaces. And got to know Tony and Ted and one of our first customers. And so what we wanted to, we've been working with them for years and stair-stepping or slowly kind of increasing the complexity of what we're doing with them in these little cube labs. And so the ExoLab that is still on orbit right now, unfortunately it was removed just a few days ago, but still in space, with everything going on, still coming home at the same time here in just a few weeks. And then already planning for our next ones, which I'm sure not sure if they've talked about quite yet, but what those future missions are. So what our focus is and what Drew might talk about a little bit, and I'm sure Ted's already shown in those ExoLabs, we basically have space versions of these ExoLabs that have to meet certain requirements and things like that to get it and operate it on the International Space Station. And so we fly those, we operate them from our ground station here in Lexington, Kentucky, and download the data, set lighting parameters and other things like that, and then obviously feed that to Magnitude's work, which is in really kind of showing all of this information on their portal. And of course, what they're working to do, obviously, is to have these experiments on the Earth run at the same time as these ones on the on space as well. So that just kind of gives you the background. I'll give it back to you, Ted, if I missed anything or anything else to talk about, or whatever questions. But hopefully, I kind of gave you guys a little bit about who we are and how we got here.
Mm-hmm. Well, I guess what's exciting to me is really the transformation that your company is able to provide in industry and in education through these labs. I actually met you, I think, through Professor Bob Twiggs. And that's how it got started. It took us a couple of years. And I think it might have been 2015 where we did our first video conference session with Dr. Ron Shigeta about growing a plant, Arabidopsis, in orbit and what some benefits might be. And really, I think we're still pretty naive about the whole process. But kind of one thing led to another. And our first mission went up with you in February 2017. And now here we are getting ready for mission 8. And so you've enabled really schools around the world. We have schools in Kenya and South Africa and in Germany and Canada, here in Japan now, as well as in the United States, that you've enabled through this architecture that you've put and through the work that Andrew's done. And so this is really kind of a new future, right? When we think about taking this vector of gravity away from the equation, if we think about biology or material sciences. And if you can speak a little bit about to some of this edge of discovery that you are really right. I know some things you can't talk about working with some of the pharmaceuticals or what have you, but you can talk about perhaps disease solutions and maybe about processes, like growing organs or organoids. This is very fascinating stuff. I know you can't speak specifically to the experiments, but in a more of a general sense, as the scientists and engineers are coming to you with a design challenge, like here is the thing we want to study, and here are the constraints that you guys have in these modules. Maybe talk about that, and then also the systems themselves, which are really fascinating. The International Space Station, this amazing platform, it's been up for 20 years, and how the lockers are used and how you're able to leverage those capabilities. So my mind just goes all over the place, I know, but why don't we start with just a little deeper about the broad field of investigation, maybe picking a selection or two. I know you even flown like Budweiser stuff. It's like everyone's interested in thinking about what the solutions are.
Yeah, sure. And was that to me or was that to Drew?
That was to you, Twyman.
Oh, okay, yeah, just making sure. Yeah, so so kind of two questions, two things to expand on there. One is the type of investigations, and the second one is how do you actually do this? And so I'm going to take the second one first. And what the big challenge is not designing for zero gravity is difficult because heat flow is different, fluid flow is different, some other things like that I can certainly talk about. But really, it's designed for automation, and what I would say is kind of long-term automation that's beyond, that's kind of beyond, just a few hours or days. This is automation for, a week or a month, a couple of weeks or a month, and that just is. That design, that is really the toughest engineering design problem, is designing, whether it's a plant experiment or a cell culture experiment or anything else like that, setting it up and it running in an automated fashion where you cannot go physically touch and fix something, where you can only command it through software. That is really the tough challenge there. And on the biological side, there are kind of these cloud lab companies around the world, or the cloud lab companies that are really starting to pop up and are trying to kind of perfect some of this. So that's really, the largest challenge. If you kind of back out and you look at our website, or you look at, other companies who do this type of works websites, you see that these are, there's a lot of kind of cube shapes. There's a lot of kind of common shapes, and that's really based off of when in the space shuttle days, they had these basically storage lockers called mid-deck lockers, and they're about, if you kind of take maybe a normal microwave and to put them, kind of one in front of the other, that gives you about the dimensions of these things, and so those are kind of how volume is partitioned, and then power and Ethernet data are provided to these volumes. And so we built our own, what we call the Tango Lab, and that's how we basically effectively plug into the space station itself. You have to think about the station. It is a spacecraft, but it's really a laboratory. It's a short sleeve environment that just doesn't have gravity. And so, and then of course we designed these cube labs that go in there and kind of these, our one use are cubes, and then of course as the volume increases, they kind of get rectangular and other things like that. So that just kind of talks about how we designed our products to interface with a human-tended spacecraft, and a few of the engineering challenges. So then if you get into, I'll take kind of brain organoid work that we've done before. For those that don't know, these are basically a kind of a clump, if for lack of a better scientific term, of brain cells that are maybe just a few millimeters or less than a few millimeters, that you can see them with your eye, that kind of emulate the brain's function at a very simple level. And I'm not a scientist who works on this, so you'd have to reference. But what we did there is, like many cell culture experiments, you've got to keep the cells and showing a video here of what these cubes look like as they're going into our Tango lab. And so I'll talk to that here in just a second. But on these organoids, you've got to keep the science itself at body temperature, 37 Celsius. You've got to keep the media, or what you're feeding those cells, you've got to keep that cool, because there's growth factors and other things in there that degrade as it's warm. So you want to keep that fairly cool. We shoot for below 10 degrees Celsius. Sometimes you might have agitation so that things don't clump together. You've got CO2 control. Obviously, you have imaging. You want to be able to see what's going on. And then finally, you have typically some sort of fixative. And this is kind of true across all biologic experiments. And so that fixative goes in towards the end of the experiment. And then, of course, it's put in a refrigerator and returned home. So that kind of process flow talks, gives you some, some idea of what integrating a brain organoid payload might be like. And just real quick, what you're seeing, I believe this is Christina. She just got back from almost a year in space. That locker you can kind of see there shows you kind of the volume of those express lockers. The Tango lab itself we built and designed ourselves, and she's closing the door. And so what you saw a little bit previously was her taking an experiment that had the Magnitude logo on it. She plugs it onto one of our cards and then inserts it in there. And then what she's doing right now is she's taking basically power and data cables from the space station. She's going to hook those up, put the on button, press the on button, and then we're ready to go. So that just shows you. And once all the computers and stuff boot up, then you're able to command and talk to these modules from our ground station here in where our headquarters is based in Lexington, Kentucky.
Twyman, can I ask a question here? This is Dan Barstow.
Yeah, sure.
This is wonderful what you've invented and your persistence in getting it to happen. So congratulations on all that over all these years.
Yeah.
I'm thinking about the changing nature of doing an experiment on the space station, where the old model is it takes years and a scientist works and finally gets their sole experiment up there, and then they take it operates and that's it. Now we have so many people engaged, and I'm thinking now on the education side. So folks like the Quest Lab, now, they have an operational experiment that kids can write code. They send the code up to it. It does the fans and the heater and the sensors and then downlinks the data. So now instead of it being one project over 5 years for one scientist, it's thousands of kids who can interactively participate. And I'm wondering about your thoughts about how Tango Lab enables many people to participate in experiments. That's just one example, and Magnitude.io Exolab is another example. But tell me your thoughts on that.
Yeah, sure. So I think what we tried to do with this, and not to speak to as just an ad for us, but what I want to kind of talk is what we've really tried to do is integrate sector, technologies that go across sectors that have allowed both here on the ground, but also in space, people to develop experiment. It's really just kind of an offshoot, a little bit of the maker movement, in terms of 3D printing, low-cost circuit board design, low-cost sensors from companies like SparkFun or Adafruit or others, have now allowed people to really design their own kind of, first it's kind of, you can build robots and other things like that, but then start to do, biological centric experiments. And so what we've really tried to do is kind of create a standard that people could then, and when I say standard, here's the connector, here's the power and the data communications, and as well as, the box it fits in effectively, for people to really develop inside of that. I think what we found, in our initial kind kind of, as the company was really getting started, we thought we would kind of open source this as this design, this design standard that people could just kind of play with. What we found though was that we really needed to be working with companies who were then interfacing with that direct customer, particularly on the education side. And so that's where obviously Magnitude has built one part of their business is in kind of coming up with the types of experiments that really had impact to their student base, and then developing that in tandem with Space Tango. And so the next thing, and just to brag on the Magnitude team, that the portal that they've built that pulls in data from what students are doing, as well as from the data that we're sending them, and kind of building that side by side portal is really helpful because it brings in what's in front of a student, as well as what's extraterrestrial, literally, brings that right in front of them and allows them to really experience space in a way that would have had much longer timelines. So hopefully I got your question there, but I think what we've tried to do with partners like Magnitude is really lower those barriers, both in terms of cost, but also in terms of just timeline. I think in like many things in our world today, and particularly if you're working in the education space where you've got I believe a 9-month teaching calendar, but obviously a little shorter right now. You've got to be able to have impact, quickly, not wait, as you kind of talked about in the more traditional sense, 2-3 years to get something on orbit. So yeah, this is perfect. Yeah, this shows and of course the Magnitude team can talk about this integration here, but this is great because it shows left and right what's happening in locally, and then what's happening in space.
Thanks.
As a reprise here, I do like to divide it at the Kármán line, Twyman, and kind of terrestrially here on the left-hand side are the exolabs around the world, students running these experiments, and on the right-hand side, Twyman with his team, like Drew Diddle, who's on the line with us as well, design this experiment, run the trials and tests, and then get our plants in gravity. We're ATLation, I'm going to get that word eventually right. We can see what's happened to this plants on the right that are on the space station. This is the current mission that we just completed. They were sitting on the pad for a couple extra days and trying to grow toward the light. So they were in the gravity vector there, sitting there at Wallops, waiting to go up. And so they just started growing and looking for light, naturally kind of going opposite of gravity. And when they finally did get up to station, they just didn't have the oomph we were looking for. But what's great with our platform is that students and teachers can monitor this day by day activity. This little slider here can kind of take us back in time. And Twyman and his team at Space Tango have enabled us to get a telemetry down multiple times per week, in which we get carbon dioxide readings and temperature and relative humidity, the light levels measured in micromoles per square meter per second, so we can get a really good sense of the lighting on the station as well. And then his team's able to also even send corrective measures if we want to readjust things or reset things, because it is a remote operation. And this is all, I think, a learning process. We're iterating along with our teachers, whether it's a pedagogical revision on how we might want to introduce this as a classroom experience, to a mechanical experience where we're just going to need to make it a little better or do something a little different. And it's a constant evolution. Dan, I think you brought up something pretty fascinating. So we fly these about 2 missions a year right now, these labs going up there. And it's pretty expensive to get the ride up and get back home again. And so we are investigating now this idea of a semi-permanent classroom in space, if you will, that enables more of a physics-based investigation, moving a little way from plant biology. But I'll tell you, with Dr. Massa, who joined us at the beginning of the day at around noon Eastern, and she spent an hour with us, and at the beginning of the video here, if you're watching it recorded, and she was talking about some of the physics of water and the physics of air, and how it's different, and how the plant has to respond in those environments. And really, the challenge then is the materials that you're using, and they're very inorganic materials. If you're looking at the ponds solution for providing nutrients and water to the plant. This current evolution of what's up there right now. We use this agar medium, which is nice and comfortable for us. It gets us up there. It's great for seed germination, but they're using like Teflon and Kevlar and Nomex fabric to grow their plants in. And guys, I know we have teachers that are excited about this and students that are very interested in this. Again, high school, university, start thinking about materials and things about how microgravity might affect fluid delivery and nutrient delivery. And it still hasn't been solved. And I think they're doing a reasonable job. I think we're doing a reasonable job, but there is so much room for improvement as we think about life beyond Earth. But I guess that's just a part of it, right, Twine? Because we also are now thinking about these plant behaviors and what we can do to make life better here on Earth. And Drew, I guess that's a great chance for you to jump in here because you really are helping us maintain our plants to the highest order that we can. And as we begin thinking, not just at the macro view, but last week we had Dr. Richard Barker join us, and he was really looking at the micro view of the genetics and how the genetics might be modified. Can we identify plant proteins, behaviors that epigenetically would enable us to maybe have a plant that lives in a saline, kind of more of a brackish water? Can we have plants that tolerant to heat or produce more crop? And so these things for research, like what you mentioned, Twyman, these brain organoids or, yeast and these other investigations, microfluidics, there's so many different ways to explore. I guess the first way to start is take gravity out of this equation that we're all so used to. Everything we're basing civilization on is this, the 1G that we get from Earth. And what happens when we have 1/6 gravity on the surface of the moon, right? Now, do you have any work or plans, Twyman, about partial gravities? I have seen some other devices that do this, like TechShot's got some stuff, I think. Or
Yep.
are you really pretty much looking at the lab as a microgravity environment?
Yeah, sure. For us right now, just a microgravity environment. I think, we've really looked at, and really TechShot does have a great machine, a company, that provides the service kind of like us. I believe it's called a MVP multivariable platform, maybe. I might be missing something there, but, they have a system that has a centrifuge that's rotating. And of course, with basic physics, you can, depending on the arm of that centrifuge and its speed, you can create a force. One of the things of the limitations, though, is that you have tidal forces. If you're doing something even over a couple of inches, and what that is that if you're 9 inches away, you might be at 1G, depending on a certain rotation rate. But if you're a quarter inch in, you might be feeling, 0.95, and then another point, and that depends on a couple of different variables. And so, the machines that are up there are as far out as they can be, and they've designed their experiments to all be kind of at the same distance away from the axis of rotation, so that everything can get that same inertial force. For us, our focus is really on these applications where we know microgravity has a value add. On the biological side, it's a little more needed, those kind of controls. But what we've really worked with some of our customers is running. Sometimes we run asynchronous, sometimes we run synchronous controls, where we build up the same hardware. One drives out to the pad and flies, and the other one stays in the lab. It's not perfect because you still have, well, what was space and what was the 4 or 5 Gs you're pulling on flight, and the Gs you're pulling on reentry. So we have to work with each different customer to see if they feel comfortable with that. We've looked at centrifuges that are large enough that can also go on random vibration tables that can emulate what that inertial and random vibration force feels like. Haven't had to do that quite yet. So there's just a number, but no, we are not looking at adding a facility of that type yet.
Well, I mean,
unfortunately, I have to say, I am going to have to step off here.
Yeah, I saw that. I took you over a minute there. I was trying to keep you right on the spot.
That's okay.
So thank you so much for joining us. I know how busy things are, especially with having now your distributed staff. So Twyman, thanks so much. I'll catch up with you a little later. Right?
Thank you, Twyman.
Thanks. Yeah. And I'm going to pass it over to Drew. Drew, thanks for joining us.
Hello. Yeah. Yes, no problem. Glad to be here. So yeah, I won't be as good of a resource of information as Twyman was, but I'll do what I can.
I bet you will be. Drew has been really irreplaceable as far as helping us figure out what we need to do, not just here on Earth, but get it ready to fly. And so I want to switch the conversation around the hardware and the operations and how that all works to really the life, right, that we're putting together. And so, Drew, walk us through, if you can, what goes through your mind as you're thinking about this, the plants and the habitats we're putting them in, and some of the testing that you go through. And man, I wish you could be in the lab right now, because it's so cool to walk through your lab. You could turn the camera on, you could show us some cool stuff, but we're going to make the best of it. I'm glad that you can join us. So share a little bit maybe about your day and what you think about when you go into these projects. It's because we always come up with the next new thing, right? And we've got our plan for October on SpaceX 21. We told you we're going to fly a legume. We have some of our teachers that joined us today. They're helping us along with anyone that's joining in. We've got this Leguminaut challenge. I think Mr. Wilkinson's there got a little logo, our next patch. So we're not quite sure which legume we're going to fly, but we're going to have to run a lot of different ground tests. And part of it is going to be having our teachers and students help us identify which candidates they think they'd like to send up. But I would like to turn it over to you and maybe a day in the life of an aerospace engineer focused on plant biology. How's that?
Yeah, I guess I'll start by saying that if a lot of the stuff that we're doing currently is kind of based off of our first experiments, which we kind of had to learn from trial and error and everything. As far as picking the plant, one of the things that I like to think of first is just the general survivability, the durability, the hardiness of the plant in itself. Of course, germination rates. We want something that's going to make sure that it's going to germinate, it's going to go strong, it's going to be able to handle some of the stresses. Like for example, I believe its scientific name is the Vinca uncuculata, but the cowpeas. For an example, like we were looking like at strains of them, such as like Texas cream and stuff, because they had a slightly higher heat tolerance, and that's one of the things that's kind of hard for us to control inside the cube labs, is something like the heat. Like it can vary. We have an average. We can of course go back and look and count, but just things along that course. Can they grow? How much water do they need? Can they survive? Basically, can we grow them in the auger like we've been using? Yeah, so just general things like that. So there's a lot of research that goes into it, from the get-go at the very beginning, before pretty much anything even gets, tested and stuff, to begin with. And then, like I said, if we were in the lab, I could show how many, just how many tubes of plants I had growing beforehand, just to try to get them to work. And, of course, we ran into a problem. Like the first source of seeds that we had that had a pretty low germination rate. So we had to switch and find the ones that you guys finally found and used. And those worked great. They worked perfectly, so. Oh, hello, Michael. See, is that a bean plant there?
That is one of the mature ones. Yeah. I don't know if you can see, it's actually produced some pods.
Yeah. Last week, wow. Michael went through a dissection looking for the root nodules. And Michael, to just recap everyone.
I feel like Artie Johnson here. Very interesting.
I guess we can reference back to your video on last Friday of why we're actually doing this particular series of experiments, right?
Yeah,
this symbiotic relationship here.
Right. So symbiosis is, Ted, I like the way that you've described it, and that it's like a barter system. So the plants are like all plants. They're photosynthetic, so they produce glucose, and the rhizobia is able to take the nitrogen from the atmosphere and convert that to ammonia, which then can be converted into nitrates and nitrites that the plant can use. So and then the plant is giving the rhizobia the carbon compounds, the sugars, and so that symbiosis is really important. We're looking at it in terms of a potential for a biological solution to fertilizer.
There's the lab.
Yeah, so these are Drew's specimens. I'm ramping back up again. I could swing around here and show you my home lab. Moved everything from school with the shutdown. And oh, sorry, that's not good. I was going to show. There we go. So we were talking about the nodules. So these are our nodules that formed on the roots, and it's a result of an infection with the rhizobium bacteria and the plant's response to that. And then that symbiosis develops inside those nodules.
Now, Drew, you mentioned, this variable of selecting the right source of seed. That's super important. But you've also been working with us to develop the right agar medium, concentration, the consistency of it, lighting, right? The conditions of lighting. So there's multiple variables here. As you work through the process, what are some of the challenges you found to find that kind of the right composition?
Well, it's an interesting question. Guess gonna have to go back and say that a lot of this is stuff that we figured out from previous trials and everything, and of course like research just about the plant itself. Like how long will they require the dark period to germinate, which in this situation, this specific circumstance, we did pre-germination on the ground, which that's makes things like quite a little bit easier. So as far as like lighting and everything, we have to really focus on like you were saying, like the heat and stuff like that. If we have to generate a lot of powerful lighting, basically like for the photosynthesis to take place, then we're also going to be generating a lot of heat off those LEDs, and we'll have to figure out a way to basically try to shunt that away from the plants, and to keep from overheating them. In the same vein as that, if we get too hot, we can actually cause condensation and start to lose moisture, because one thing we like to do is, we like to keep, basically we like to seal the top of the so where our gas sensor is actually external to the tubes that the plants are in. So we like to have basically a gas permeable membrane there at the top, so that we can have gas exchange throughout the inside of the cube, the internal compartment of the cube lab, and within the tubes where the plants are growing. So that's how we can monitor the different gas levels and everything. So we can actually cause like condensation on the tube and like obstruct video. Like we have to think about everything, down to that level, to make sure we can get images and everything. So yeah, I mean, if you'd like to ask like more specific questions, I can try to go into it.
It's that balancing, act that you're helping us walk through, as you mentioned, too much or too little. It's kind of like the Goldilocks equation, right? It's got to be just right. And one thing I never really even considered, imagine, if you will, being on the space station. What does it sound like up there, right? There's a certain cacophonous background noise of all the equipment humming, and so there are sound thresholds that are put in place for our labs, right? And we need to move air around.
That's correct.
And so some of the first fans that we were running, it was like, those are too loud. It's like, oh, wow, but we have to push so much air through. So we had the constraints of things you didn't even think about, like sound, because you want a nice operating background for the astronauts, because they got to live in it for 3 months, 6 months, a year. And then you have for the plant itself, and you still have to move the air around. The plants, they need the right amount of light, right? They need the right amount of kind of exchange, and then the medium that it's growing in. And so it's not just a variable of like, let's put something and see what happens in microgravity. We have a lot of constraints within this engineering challenge of 20 centimeters by 10 by 10 and making something live in that. And the fact that we're able to do that for a number of weeks is quite fascinating to me. you'd mentioned something I think was really important for these specific experiments, the pre-germination, in order to kind of hope that we can kickstart the root, develop the nodules there at the roots, because that's really what we're looking at, right? We're looking at that nitrogen fixation. And is that something that's replicable? So, looking at the other plant experiments, is there anything you can share from your knowledge of this experiment, or something we might want to take in consideration here? Everyone that's immediately here in Zoom is a teacher or will be directly involved in the process of determining this next flight with some legume. But what are some maybe things that we've learned from this last flight? And I think it's okay to be a little transparent there, as long as we're not talking about money, right? But the engineering challenges and what have you.
So
those challenges that you think we're going to need to be facing looking at the next flight coming up, which sounds pretty similar.
So one thing that I definitely think we'll need to look at is, I don't know if you guys noticed from the picture that he put up a few minutes ago, but one of our auger tubes, I guess on launch, the auger had scooted up from the bottom of the tube, the Pyrex tube. So I was actually trying to think about what might have caused that in that particular tube versus the other ones. I was thinking of things from the fact that it may have been like when it was sitting on the pad for too long before launch, that the root growth, it likes to get a little leggy when it doesn't have light in those first stages when it should have it, may have grown to the edge and kind of broken the seal in between the glass and the auger, making it easier for it to scoot up on launch in that particular tube. So that's one thing that I'm going to look at and try to make sure it doesn't happen in the upcoming flight. So, and as far as how to tackle that problem, haven't really gotten there yet. It could just be that, making sure that we have a better system in place for a scientific refresh, even if it is sitting on the pad for too long. But those are questions that we'll eventually be tackling.
Well, why don't we open it up and see if any of our teachers have questions? Clearly the exolab that you have in your classroom is different than the lab we send to the station. So, any questions that our teachers have here, please weigh in and ask them.
So I see one question in the chat. Let's see, student question. Seeing if plants could grow upside down in space since they don't feel gravity. Okay, so it was an idea on the auger moving up. The question was, what if we could somehow do that on purpose and grow a seed out of both sides of the auger? So the only problem that I see with that is yeah, it would be basically the same in microgravity. There is no up or down, like there's no against the gravity. So growing it on either side would technically be 100% doable and achievable. The only thing I would think is that we'd have to have basically a tube with open ends on both ends, because the seed that's on the bottom, if the auger sticks and does its job properly, would be restricted from airflow. So if we could open it up on the bottom and somehow think of just an open-ended cylinder, that's something that we could possibly do. But then we'd run into the problem of trying to fix the auger in place there, because it would be best if we could basically have the auger not move. That's how it makes the best seals. If we had to fix it at one end and then flip it sideways and push the auger in, that would just inherently kind of ruin the bonding between the auger and the glass that we need to help keep it from rising up or moving on launch. We'd have to fix it there in place and possibly even launch it sideways and have it require descent orientation. Just a couple of things that come to mind. It's a good idea. It would vastly increase the amount of space that we would, or I guess technically usable space that we would have inside the cube labs. But it would also present some rather interesting engineering challenges we'd have to overcome.
One thing that I've been thinking about is our plants are kind of cheating on us a little bit. We want them to study them in microgravity and what happens with, like, say, phototropism and gravitropism. And we don't have omnidirectional light, which is pretty hard to do. But if we could totally fool a plant where it didn't know which way the light was coming from, because it was coming from everywhere, then what would happen to root development, right? And so right now, I think if you look at pretty much every experiment that's on station right now that's dealing with plants, has a light source coming from a single direction. And so the plant's kind of cheating. It's going, well, that's the way the light's going. So if the light's going this way, this is my a layperson's guess on it, then the roots must be wanting to do the opposite thing. And maybe we need to get our plant biologist back on here again. So one challenge, and I don't think we can get that in a 2U lab and keep the temperature down and it quiet enough. And right. So that is, you know, when we think we're just studying about plants, there's that whole question around engineering practice. And for any of you in states with the next generation science standards, when we're dealing with the science of the plant growth and microgravity, but now how about the engineering practice? If we had the luxury of the veggie box, of the veggie that we saw up there with Dr. Massa, then we're in the open cabin. They even have those little accordions to kind of move up, a little shield, if you will, so they can keep that airflow going with the fan on the top. And they don't run that all the time, I guess. So that's kind of interesting, right? That we have to consider the constraints we have from an engineering side. And Drew, you know, it's been invaluable for you and your team to help us walk through some of these problems. And if your students have ideas, or if you as a teacher have an idea, it's worth bringing up and having a conversation about, because whether or not it's a solid direction to go in, I would love our students to have that curiosity to imagine how they might want to solve these, what appears almost intractable problems of trying to do so much in so little volume. You know, for me anyway, this is pretty exciting.
Drew, I have a question.
Michael,
I can't believe that I never asked this along the ways. Do we have a known orientation when we're loaded on the spacecraft?
Yes, whenever we're loaded on the spacecraft, it would be as if the plants are sitting in the tubes in your lab. So they basically they're growing up when they're on the launch pad, and that's, you know, a couple of reasons. You know, one, to try to help that auger stick to the bottom. One, so we don't have, you know, basically that's also the direction in which the auger is the longest, and as long as the roots are in gravity, I imagine they're going to be exhibiting some gravitropism, which that's actually one thing. You know, go back to Ted's comment earlier. That is one thing to think about. Is so far what of our experiments have been doing, they've kind of been not just eliminating gravity but isolating phototropism. So that's one way to look at it, and just, you know, kind of think about it in those terms, which would kind of make your scientific focus a little bit more straightforward. You know what I'm saying? Like if you can look at it and just say, oh yeah, well, these effects are the result of phototropism. So
right.
back to
raise that question. Go ahead.
I'm sorry. Yeah, no.
Go ahead, Drew. Okay. I was going to say, what makes me raise that question is just that conversation with Dr. Barker last Thursday, in which he's really kind of looking at the genetics and really very specific sequences of these proteins and how they're affected by very specific things. And we have all these variables we're throwing at the plant, and we're trying to identify specific things. And it's when we have these variables, again, and this is true not just with our experiments we fly in the labs that go up there with Space Tango, but in all the experiments that are up there. They're trying to reduce it down to a single thing that they want to investigate, I would imagine. But the more you add, it really kind of complexifies. It makes it more complex in terms of trying to tease out what's exactly happening. And so, I mean, we're on a path of learning as we build this out. Tony and me and our team at Magnitude, Michael, as we build this out, we're learning with you. And it's really, we really literally are at the edge of discovery and how we can make these systems work. And I think us being more transparent, if anything, by having sessions like this, helps me realize that we should probably be more transparent to you as educators. And if you're students out there, for you as students to be thinking about, no, they haven't figured it all out, and they're still trying to figure it out. How will we grow plants on the surface of the moon? No one has an answer for that yet. Even our most advanced folks that we bring in here that are working directly with at the very edge don't have those answers, those questions answered. They're still trying to figure out what these plants do and the behaviors of these plants, whether it's looking at a model organism like Arabidopsis, or these legumes that we're trying to find that we want to fly because we want to create a green fertilizer perhaps in the future. So the future is yours, students, as you know, whether you're 8, 12, 18, 88. I mean, we're all students, right? So it's a chance, I think, for everyone to get involved and think about what they can do. We like the narrow focus because we can then pedagogically walk through some very specific things, but keep in mind, it's very open-ended. This is not a classroom setting. These are not bells ringing every 55 minutes, right? And so we have a chance to explore, and even more so now with the uncertainty, and we're at home and we're doing things whether we're teachers or students. So I think we can have a lot of fun as we imagine what ExoLab 8 might look like. Keeping in mind, we've got engineering constraints, and we can't reinvent this thing. We can't make it, 60 centimeters, but whatever, by 60 or whatever. It's got to be within these constraints. And I think the best design thinking happens when you do set with constraints, rather than having a whole bunch of things you can do. So what can you put in a 20 centimeter by 10 by 10 with lighting and power that enables the plants to grow prodigiously, we hope, in these microgravity environments? And I think we've gone through 7 steps of evolution. We're going to do it in the 8th with some changes. So I don't think we're seeing any major changes today, but we might. We've got a little extended time now with the launch in October rather than August. But from an engineering perspective, putting in just kind of thinking about it, you really need upwards of a year or longer to really think about changing your hardware in any significant way. Changing a plant design and what have you, it's fundamentally the same kind of plant, or we're looking at plants versus saying a brain organoid, then we can kind of keep everything pretty much the same and think about just swapping out the test candidate. But for the hardware, to your experience, Drew, I'm sure that you can't just suddenly say we're going to build this whole new thing, because how many failure points are we going to have? So it's just the constraints, and I think that's the exciting part, that we do have constraints, right? Is there anything that you might advise us as teachers, as junior researchers, if you will, on bringing design and ideas to you? What makes your job easier?
So I would say one thing that would definitely makes our job easier is whenever you're thinking about the design of these experiments, keep things in mind, like we're having to deal with problems that basically, like for terrestrial experiments, they've been solved for hundreds of years. Like, for example, how do you decant a liquid in microgravity? Like that's such a simple task, if you think about it, like on Earth, but then it just doesn't work in microgravity. So it's think about the small things. I guess is what I'm all I have to say about that. It's just, that it's the small things. And I also have to say that exactly like you said, we don't have everything figured out. Like, for example, the double-ended tube that was just asked about. Like, that's an idea that I hadn't thought of before. And it's just things like that would basically really I guess increase the brain pool.
Oh, there you go. Maybe if we thought about more ground cover type crops, right? They weren't really leggy, if you will. Then we'd have the volume in which we could do something on a dual-sided, multi-sided. You have a sphere that's floating in microgravity, and everything's kind of, it's like there are all those ideas, and how do you actually implement it? Dr. Massa,
Lori, would you go ahead and throw in your thought here? You just put it on the chat.
Oh, Lori, yeah. What does Lori say here? Lori, go ahead.
Well, what would prevent us from, if you want to do like the ground cover, what would prevent us from turning the ExoLab on its side, and then you have an increased surface area sideways, lengthwise, for the long dimension of the ExoLab? And then you could also see, is it indeed the light that's drawing it one direction or not? You could even do 2 plates, this way. Have them I don't know if you'd want them to grow towards each other, but have them on the exteriors of the box towards the center. And then you could do the same thing in the classroom. You just put it inside the sleeve, and that means it's limiting to only the light inside the box and not getting classroom or window light. So that's maybe a way to do multi-dimensional things that you could duplicate on Earth.
You've got me, my neurons are firing here, Lori. What we can do for fun is have a series of design ideations in which we open it up, we talk about what we're doing, and people can just riff, and we can just take notes. And if we want to do trials, anyone that has an ExoLab can do those. I have a clinostat. The clinostat is this, basically it rotates the object, in this case a plant. We're using a single axis clinostat. The plant gets confused, it revolves once a minute, and it doesn't know which way the gravity vector is. So pretending like we've got a microgravity environment for the plant down here on Earth, so we can do things with that. Of course, there's nothing better than getting in one of Drew's boxes and getting up to the space station, though, right? So we're getting close to the top of the hour. So if we can, Drew, thank you so much for joining us and providing a little bit of insight. I'm going to guess there's going to be some questions as our teachers are thinking about this that will end up popping out to you. And Michael, if you can kind of just cap it all up with our 3-hour session here, with what we're actually doing with our Leguminaut Challenge. It's coming up in October. And everyone that's been watching these and our teachers that are actively involved have a chance to actually help us determine our next candidate to space. And it sounds like if we get into this a little bit, that we might even be able to work a little bit on some engineering design questions, keeping it within reason, because I think, as I mentioned, there's some pretty difficult things to do to make sure that stuff works. And we don't want to make some major changes unless we're planning for something maybe in the fall of 2021. If you want to think about redesigning something fundamental, then that kind of puts your horizon out there at least a year. But for now, we're kind of working within these constraints that we have. We can do little tweaks, might change the lighting, the actual LED lights that we use. We might try a different one. Dr. Massa talked about white LEDs and how they haven't even really been used up there. And the red, green, blue that we see up there is because that's always been up there. You know, that's funny. That's because that's what we're using, because that's what they're using, but they haven't looked at the latest stuff. So maybe we actually have a chance to advance just by going to white LEDs. On station, it's going to be harder to do here on Earth, but you get where I'm going with that. So Michael, why don't you wrap it up for us here and give us a kind of a summary of what we're looking for with ExoLab 8. And Drew, thank you so much. And thank Twyman for us that you guys were able to pop on, as well as Janelle. Awesome. Thank you so much for your inspiration. And earlier, Dr. Joya Massa with her insight on that kind of edge of research on creating food for our astronauts. So why don't you cap it off for us, Michael?
It's been an incredible 3 hours, and it actually has flown by very quickly. So if you're joining the end of this, go back and watch it from the start. I know I'm going to go back because there's some things I want to make sure I document, but so many good things to think about in terms of the legumes that we're selecting. Drew's comment here, just the little things that seem so easy here in 1G that become really complicated in micro G. So that's kind of the whole point of where we're headed here. And you know, in Janelle's words of just give it a shot, see what happens. You know, what's the worst? You actually learn something. So, and I always tell my students is if we got it right the first time, we wouldn't have learned anything. We seriously would not have learned anything. The process of going through this and having these failures, if you want to call that, but different iterations of and different trials, we're learning so much more about how things do and don't function in the microgravity environment, in the elevated carbon dioxide levels, the temperature levels, the changes in the loss of the weak force of gravity and the strong force of surface tension taking over, and how our plants can actually drown because the water's coated the roots and now they can't get any air. You know, we've got these bubbles of carbon dioxide around wrapping around the leaves of our plants. The surface tension, think about that, the surface tension of a carbon dioxide. The gas has surface tension, and we would never be able to think about that in 1 gravity because the force of gravity on those molecules is stronger than their adhesion to the leaves of the plant or the roots of the plant. So there's just so much here that you can head into, and really what you know what's mentioned with this Leguminaut challenge, we're trying to decide what the next best steps are with this investigation. We're trying to find out, can legumes nodulate in space in a habitat off of Earth? Can we fix the nitrogen that we need to crop other things? Those microgreens that Drew, that everybody's spoken of, that, and you know, we have a couple of microgreen experiments as well that we've done. Those are all nitrogen intensive plants. And nitrogen is a limiting factor to growth. And if we're going to grow those things that humans need for vital nutrients, for the taste components, so just so that you sustain your appetite, we've got to be able to feed the nitrogen to those plants. And the only thing that can convert that for us are the legumes with their symbionts rhizobium. And we don't know if that can function. There's been a few experiments that precede us that we've built on what they've tried to do, but nothing's gotten far enough to be certain. There's, as Drew just mentioned, there's so many tiny little variables that we've got to look into, and that's the true experimentation. So I really, we're inviting all you learners, teachers, educators, thinkers, designers, artists, everything.
The planet, 8 billion people.
Yeah, your input is valuable, and we want to hear from you, and we want your ideas. Contribute to this greater project that is going to solve some problems. Liam earlier mentioned, everything we do in space is really we're doing it for Earth. And the ISS, its theme is off the Earth for the Earth.
There you go.
And we're going to have to figure out the sustainability issue on this planet. We're going to have to figure out how we're going to cope with elevated carbon dioxide levels, how we're going to cope with warmer temperatures, how we're going to cope with less moisture, all these same issues that we're struggling with on the space station. It gives us a little micro environment that we can manipulate a little bit more and learn, so that we've got some solutions for here. So please go to Magnitude.io, join us, have your students join us, come on board, share your ideas, adopt or pitch a legume. Tomorrow we're going to have presentations from 6 of our team leaders, and then we'll have a few more next week.
Absolutely. So keep in mind, we're really thinking about you with your students, or if your students being at home. Hardware and computer internet access is not important to do the design challenge. We need your imagination. Younger students, send us, we put our mailing address up there. I'd love to get a picture or a drawing or a postcard with your ideas. So it's okay, just write it down, share it with your teacher some way. There's ways for us to do this. I know we can solve that last mile, if you will, of knowledge. And for your advanced students out there, go ahead and look at some of the white papers. Look at what Dr. Moss has done, or Dr. Barker, the research that they have done, and build on that research. You don't need to go into the forest without any of the equipment. These folks have already been out in the jungle. They kind of know this jungle of knowledge, and they've kind of carved some paths you can go down, maybe make your own path, but it's there for us. If we're lucky, we might even be able to get someone from Genes in Space to join us. They've been doing really some interesting work as well. It isn't a requirement to have hardware or software, or even to have a lot of scientific background. Bring your imagination, bring your creativity. Our teachers, our educators here will help us, help you determine how you want to join us. So thank you so much for joining us again today. Tomorrow, I think it kicks off at 11 o'clock Eastern, 8 o'clock Pacific. And we're going to have, I think about 4 hours, but most of it's going to be about learning about these different legumes. We're going to be sending up which one's the right one. And Liz Warren from the U.S. National
No, not that
Laboratory,
Liz Warren.
Not Liz Warren, the presidential candidate, but the Liz Warren who we really love, because she's studying microgravity and the behaviors on things like bones in the human body. And Mark and Scott Kelly, the astronaut twins, we're basically doing the astronaut twin experiment, except with plants. So our Scott Kelly plant's up there. Drew helps us get that plant up there. We have a lot of Mark Kelly plants, if you think about the Kelly twin experiment here on Earth, and the plants are a lot less expensive. And if something goes wrong with the plants, it's a little better than if something went wrong with the person. So plants are a good way for us to start. So guys, thank you for joining us again today. Feel free to send us an email, just hello@magnitude.io if you have some questions. We'll get you started the best way we can. You guys have a great afternoon. Stay safe. Keep your hands clean, I guess, and love your neighbor. Love your family. Your pets love you, right? Everything's like a new holiday. We'll catch up with you tomorrow, I hope. Take care.
All right, bye, everybody.